Collection of articles on HIV/AIDS by various authors. Part 2. Articles by Stefan, Lanka, Anthony, Brink, Rosalind, Harrison-Chirimuuta, Etienne de Harven. HIV, Reality or Artifact? By Stefan, Lanka. Continuum, April/May 1995. An error can never become true however many times you repeat it. The truth can never be wrong, even if no one ever hears about it. Mahatma Gandhi. For the past 10 years or so it has been the accepted wisdom that the human immunodeficiency virus, HIV, causes AIDS. It supposedly occurs in many body fluids, and its transmission, especially in semen and blood to a new host, triggers a slow but inexorable progression to AIDS and ultimately death. To infect another cell, HIV must at some stage in its life cycle exist as a separate and identifiable entity. What has been ignored and kept from public awareness is that there has never been a workable HIV test and that the definition of positive has always changed according to the views of different organizations dealing with it, changed also according to the kind of tests used and changed from laboratory to laboratory performing the tests. Its techniques have not been standardized, and the magnitude and consequences of interlaboratory variations have not been measured. Its results require interpretation, and the criteria for this interpretation vary not only from laboratory to laboratory but also from month to month. 1. The dispute over who discovered HIV, 2, was a distraction from the question of whether the virus actually exists at all. The public was impressed that if a president and a prime minister, 3, had to meet to resolve attribution, then the thing they were negotiating about must be real. In 1993 a research group from Perth, Australia succeeded in publishing a paper on the HIV test. 4. Since then anybody could have read for him or herself that no AIDS test could ever work, because HIV has never been isolated nor even shown to exist. Since AIDS research and the media have largely ignored any critique of HIV equals AIDS, especially the essential question of whether HIV really does exist, it is time to call again for a reappraisal of the whole HIV/AIDS hypothesis. In going back to the origins of HIV virology and telling the HIV story, a view will be presented which will make clear that HIV itself, the very object of this Manhattan Project of modern medicine, AIDS research, does not exist. 5. A little virology. Viruses are essentially just packages of genetic information enclosed in a coat which consists of proteins. They can reproduce themselves only by infecting a suitable host cell and appropriating the chemical machinery they find there. The proteins making up the viruses are characteristic for each species of virus. Apart from enveloping and transporting the genetic information intact, the composition of proteins for a given virus results in a specific shape for the virus particle. This much is generally known. Less well known is the existence of other particles which look like viruses but aren't, and are nonchalantly referred to as virus-like particles. Such particles are far from rare, found, for example, always in placentas, and very frequently in the artificial environment of laboratory cell cultures. They have served to muddy the waters considerably as far as AIDS research is concerned, because particles just like these have been called HIV. To date, none of these has been characterized and shown to exist as an entity which one may justifiably call a virus. One root of the belief in the AIDS virus. In classical theory, DNA encodes the genetic material of heredity, which is then transcribed into messenger RNA, which in turn specifies the assembly of amino acids to construct the proteins of all living beings. In 1970 an enzyme, biological catalyst, was discovered in extracts of certain cells which was capable of converting a molecule of RNA into DNA. This was a revolutionary discovery, because it overturned a fundamental tenet of molecular genetics, namely, that the flow of information was strictly one-way and never reversed. It had hitherto always been thought that DNA was transcribed, converted, into messenger RNA and that the reverse process from RNA to DNA was impossible. The enzyme responsible became known as reverse transcriptase, 6, and a lot of new myths arose. An error of the past, cancer caused by viruses. It was believed that the new enzyme was a marker for a virus, because the cells in which it was detected, and which were used to study cancer, 7, were thought to have become cancerous through being infected by a virus. New to the idea of cancer viruses, 8, was that nucleic acid, when in the form of RNA, could be converted into DNA by the enzyme, thus providing a mechanism for viral nucleic acid to be inserted anywhere in the chromosome of the cells. 9. These new viruses became known as retroviruses. 10. The insertion of certain retroviral genes was thought to trigger cancer. The idea that these postulated viruses caused cancer quickly became hot news the world over, but did not survive investigation. 11. And other explanations were sought. 12. The theory did not predict or explain the dramatic increase in cancer cases. Cancer could not be shown to be transmissible, nor could it suggest any remedy in the form of a vaccine. 13. Interestingly, the spread of cancer viruses was blamed on homosexuals, prostitutes and black people, just as AIDS came to be 13 years later. 14. Whenever and wherever reverse transcriptase activity was detected, it was rashly assumed that retroviruses were at work. This turned out to be a grave error, because it was later found that the enzyme occurred in all living matter, proving that reverse transcriptase activity had nothing to do with retroviruses per se. 15. Repetitive elements. Further research showed that at least 10% of mammalian DNA was composed of repetitive sequences which were referred to as nonsense genes, parts of which, nonetheless, were described as retroviral genes. They exist in their hundreds if not thousands. Some of them can even replicate independently and jump within and between chromosomes, and for this reason became known as retrotransposons. In the laboratory they can be made to migrate, and when this happens reverse transcriptase is invariably detected, which underlines the fact that reverse transcriptase activity has nothing to do with retroviruses as such. 16. LAV, HTLV3, HIV, and all that. Because all this was already well known in 1983, it is incomprehensible that François Barsinassi, a member of Montagnier's group, as well as Gallo's group itself in 1984, claimed to have discovered a new virus, when all they did was to demonstrate reverse transcriptase activity, and to publish photographs of cellular particles without proof that they were viruses. They could neither isolate them nor show that they were responsible for creating the observed reverse transcriptase activity nor the tissue abnormalities from which they were obtained. 17. They concluded, the role of the virus in the etiology of AIDS remains to be determined. 18. What makes a virus new? The isolation and purification of a real virus is a straightforward matter, because unlike cells, viruses of one species are always of the same size and shape, and can be readily separated from other cell components by standard techniques. A control experiment is to try an isolation with putative non-infected material in exactly the same way as the supposedly infected material. Nothing should be isolated in this case. To identify a virus definitively, a first and simple step is to photograph isolated particles of it in an electron microscope, and they must look like the viral particles observed in cells, body fluids, or cell cultures to distinguish them from other cellular particles which look like viruses, but are not. Proteins making up the viral coat must then be separated from each other and photographed. This produces a pattern which is characteristic of the species of virus. A similar separation and identification procedure must be gone through for the DNA or RNA of the virus. Only after the viral proteins and nucleic acid components have been properly identified, is it legitimate to speak of a new virus. No evidence for the existence of HIV. Such evidence has up till now never been produced for HIV. No photograph of an isolated HIV particle has ever been published, nor of any of its proteins or nucleic acids. No control experiments as mentioned above have been published to date. What has been shown are photographs of virus-like particles in cell cultures, but none of isolated viruses, let alone of a structure within the human body having the shape ascribed to HIV. What the whole world has seen are models representing HIV with dish areoles, said to be receptors with which the virus attaches itself to cells. The existence of HIV is inferred from an antibody test, but how this is supposed to work, when the virus has never been shown to exist and obtained free of contaminants, remains a mystery. The AIDS test. Let us recall that the AIDS test is supposed to detect antibodies produced by the immune system in response to infection by the virus. This is routinely done by layering proteins ostensibly from the virus in the wells of a plastic rack and adding blood serum to be tested to each. If antibodies are present, they bind to the proteins, and when this happens, sophisticated staining procedures can make this visible. But, because no proteins which are viral and free from contaminants have ever been obtained, one cannot be sure what the antibodies are that bind to the proteins. This is the crux of the problem facing all HIV/AIDS tests. The inability to isolate a viral entity, and to characterize its constituent proteins unambiguously means that the evidence for the existence of HIV using antibodies is just arguing in a circle. Antibodies that are detected are due to other causes. Why no HIV test is ever able to work? It is consequently quite illogical to claim that a positive test results from prior contact with the virus. 19. Because various ill-characterized proteins are involved, every test kit manufacturer applies his own arbitrary criteria, and no two kits ever give the same result. It makes no difference that learned committees set standards to decide which tests should be regarded as positive and which not, because this merely skirts round the problem, namely, to what are antibodies actually being detected in the AIDS test. It is of no help that nowadays second and third generation tests exist using synthetic proteins which give greater consistency and comparability, because only by an unscientific stretch of the imagination are they viral proteins. Neither fudging the true identity of the proteins, nor advocating two kinds of test, reassuringly but mistakenly described as search and confirmatory tests, resolves this difficulty. The ELISA test is used to screen for antibodies, which is confirmed by the more specific Western blot. The dilemma cannot be stated more poignantly than by quoting from the leaflet accompanying one such test kit. The test for the existence of antibodies against AIDS-associated virus is not diagnostic for AIDS and AIDS-like diseases. Negative test results do not exclude the possibility of contact or infection with the AIDS-associated virus. Positive test results do not prove that someone has an AIDS or pre-AIDS disease status nor that he will acquire it. 20. Quite. The direct proof of HIV. Some HIV researchers have tried to circumvent the problem by pointing to something called direct evidence for the virus. All that this meant, though, was arbitrarily selecting a protein of a certain size which happened to coincide with that shown in HIV models. The delusion of such evidence was illustrated when the protein later turned out to be of human origin. 21. How the genetic information of HIV was manufactured through. Despite this deplorable state of affairs, the majority of AIDS researchers still cling to the authenticity of HIV, because a genetic sequence for it has been published. Moreover, genetic procedures now exist, which, unlike antibody tests, attempt to identify the presence of HIV more or less immediately, instead of only weeks later when antibodies are formed. The fact that the genetic tests, PCR, 22, do not give the same results as the antibody tests is simply ignored. Since no virus has been isolated, it follows that no nucleic acid has been isolated from it either. Complicated procedures are even so described in the literature, at the end of which something is produced which is called the nucleic acid of HIV. 23. A test tube. HIV and its DNA can allegedly be made by the bucketful. 24, but under very surprising conditions which, inter alia, entail the use of extracts from plants and other oxidizing chemicals, which could not possibly exist in vivo. Immortalist cell lines devised, and later patented, by the Montagnier and Gallo groups are co-cultured with extracts from human cells or the cells themselves. At the end of it all, HIV itself is not actually obtained, only reverse transcriptase activity is shown to occur, which is taken to imply that the DNA that is found must have been viral in origin. The real explanation of what happens is as follows. In the mixture of cell cultures and stressed human cells, RNA and reverse transcriptase come to be produced in large amounts, because the cells have been specially selected and treated to do this. The RNA is transcribed into DNA by reverse transcriptase, and long pieces of DNA are produced which are said to be viral DNA. In fact, they are composed of unrelated pieces of expressed cellular RNA, transcribed into DNA and linked together by a process of template switching, a well-characterized property of reverse transcriptase. 25. This misleads ordinary researchers into believing that they have actually produced viral DNA. It is said that this linear DNA is the free or the non-integrated form of HIV, which furthermore is said to be a unique feature of HIV, because a lot of detectable free linear DNA has not been suggested in any other models of retroviruses. And a selecting process. The resulting pieces of DNA, too, are necessarily both shorter and longer than the correct length of HIV. Pieces corresponding to the correct length of HIV must be selected for size, because otherwise the purported DNA preparation would be a mixture of various lengths, which would violate a cardinal rule of virology that all nucleic acid of a particular virus be identical in size. And a detecting process. Having artificially prepared DNA pieces of uniform length, they are still not ready for presentation, because they consist of a mixture of all kinds of RNA fragments transcribed into DNA and thus cannot be shown to represent unique viral DNA. Accordingly, the mixture is subjected to a kind of lock and key detection process called hybridization, whereby pieces of DNA are detected which complement more or less a probe of that which it is desired to be shown to have been prepared. And choosing a desired probe. Since no DNA from HIV existed to hybridize with the prepared DNA, Gallo and Montagnier simply used stretches of DNA from what they said was specific to HTLV, a retrovirus Gallo had earlier claimed to have discovered, and which they deemed suitable for this purpose. The DNA detected in this way was replicated and certain stretches of it cloned and declared to be the DNA of HTLV3, later to be called HIV. To summarize, the purpose of the exercise is to grow HIV, but it actually produces a mixture of different lengths of DNA, contrary to theory which says they should all be identical, and no virus at all. It is then claimed that the correct DNA has been prepared by finding certain strands in this heterogeneous mix by hybridizing them with an HTLV DNA probe whose sequence is known and defined to be similar to HIV. However, non-hybridizing strands of DNA should not be there at all, and the fact that they are, proves that a complete ragbag of DNA has been prepared, without any indication of what it is made up of. It follows that HIV DNA must just be a laboratory artifact constructed to a preconceived idea of what retroviral DNA should be, and this assessment does not even raise the question why no virus can be obtained, whatever the experimental conditions. Gallo and Montagnier's cloned HIV DNA. One cannot help asking why no one had not long ago spotted the flaw in the techniques employed by the Gallo and Montagnier groups. After defining some segments of DNA to be HIV-specific, every researcher in the field worked exclusively with short, cloned sequences, never the whole strand, on the reasonable assumption that the original characterization had been correctly performed. From the isolation and identification procedure described above, it follows that the resultant sequences vary widely from one preparation to the next, which sequence analysts misinterpreted as the legendary capacity of HIV to mutate. A computer-simulated phylogenetic tree was constructed, which established precisely what its designers sought to prove. 26. Some history. Perhaps one reason for this calamitous state of affairs is that HTLV3 was presented to the world as the cause of AIDS at a historic press conference on April 23, 1984. A patent for an antibody test was applied for on the same day, instead of making the evidence for it available beforehand, as correct science demands. The undue haste may be explained by the fact that both the National Cancer Institute and the Centers for Disease Control, CDC, had actually one day earlier in a lengthy front-page article in the New York Times on April 22 come out in favor of the French claim for priority. 27. 2. Even so, one must admire Gallo's audacity, because using the same technique he claimed in 1975 to have discovered the first human retrovirus, HL23, but which turned out to be nothing more than pieces of DNA from 3 different sources of contamination. 28. Nowadays, Even an undergraduate would know that if you added DNA to a cell culture, part of the DNA would be incorporated into the cells without any virus being involved. What does the AIDS test actually test for? Since HIV has been shown to be a laboratory artifact, it must be assumed that, when not just cross-reacting with other known antibodies, the AIDS test detects antibodies against proteins produced in the procedure itself. They must be of human origin because the cells used originated from leukemic patients. Test positivity, logically, results from immunological contact with them. However, since positivity actually correlates with otherwise unrelated factors such as rheumatism and sunbathing, no specificity can be ascribed to the test. 29. Whether antibody positivity really correlates with disease as is commonly supposed, remains to be determined by a critical re-evaluation of the data. Condoms, therefore, serve only to protect against venereal diseases and as contraceptives, and worse, lull the user into a false sense of security by ignoring real dangers he may be exposing himself to. Redirection of AIDS research. AIDS research is therefore back at square one and not at basic science as suggested elsewhere. 30. The main players have since 1993 begun to slink off, arguing that the virus having mutated so much is now no longer detectable. AIDS has therefore to be explained in the absence of further whole virus. 31. Apart from the shortcomings of the antibody test, other misconceptions such as T-cell counting exist, which mean that the whole concept of AIDS needs to be completely revised. 32. It must be shown that there is any point in renaming a collection of known diseases as AIDS, just because someone is positive in the antibody or genetic, PCR, tests. Leaving HIV out of the picture explains why the epidemiological projections, which years ago had forecast a worldwide epidemic, have been a complete failure. Africa in 1986 was held up as a dire warning of what would befall the Western world. There, AIDS was diagnosed by a combination of clinical conditions, 33, such as chronic fevers, diarrheas, coughs and weight loss, all symptoms of the diseases of poverty, without testing for HIV antibodies. 34. It should hardly come as a surprise that an entirely different definition produced a different outcome. Finally, the effect of a positive test result on mental and physical health needs to be considered and investigated. 35. Antivirals. Whatever happens, the use of AZT and other antivirals which are supposed to target HIV replication, but actually kill cells indiscriminately, and ultimately the whole body, must be stopped immediately. It is especially distressing to note that AZT and its analogues preferentially attack those cells which divide most rapidly, namely, cells in the intestines causing diarrhea and malabsorption of food, and in bone marrow, ironically, the primary production site for cells of the immune system. 36. The people who need enlightenment. The most important and delicate task is to convince HIV positives that their test result is not a death sentence, to be generally supportive of them, to assuage their anxiety, and to help them understand that with appropriate treatment of any specific disease, they have a good chance to retain or regain their health. The large number of long-term positives, whose condition cannot be explained by conventional AIDS theory, as well as the phenomenon of seroreversion, return to negative test status, provide eloquent testimony to this. HIV/AIDS researchers and health officials are herewith called upon to debate the whole subject of HIV/AIDS openly and humanely, and to recognize the mistake that immune deficiency was acquired by an infectious agent. The future. To be able to live a fuller life, we have first to regain and then retain autonomy over our bodies and health from self-appointed experts, who have dispossessed us of it. 37. If we refuse to learn from what has happened in AIDS research and related medical policies, then worse is on the way. Some of it is, indeed, here already. 38. The genetics agenda begun in the 1860s. 39. And a primitive genetic determinism have become established through the availability of genetic sequences and the ability to manipulate them easily, which are, in fact, pure fantasy. 40. Furthermore, all models of genetics and associated technologies, e.g. genome therapy, are based on a one-dimensional,
static model of genetics which is a crass oversimplification, not defensible even when Mendel first proposed it. 41, **. Health as a virtue, Ivan Illich. Health designates a process of adaptation. It is not the result of instinct, but of an autonomous yet culturally shaped reaction to socially created reality. It designates the ability to adapt to changing environments, to growing up and to aging, to healing when damaged, to suffering, and to the peaceful expectation of death. Health embraces the future as well, and therefore includes anguish and the inner resources to live with it. Health designates a process by which each person is responsible, but only in part responsible to others. To be responsible may mean two things. A man is responsible for what he has done, and responsible to another person or group. Only when he feels subjectively responsible or answerable to another person will the consequences of his failure be not criticism, censure, or punishment but regret, remorse, and true repentance. The consequent states of grief and distress are marks of recovery and healing, and are phenomenologically something entirely different from guilt feelings. Health is a task, and as such is not comparable to the physiological balance of beasts. Success in this personal task is in large part the result of the self-awareness, self-discipline, and inner resources by which each person regulates his own daily rhythm and actions, his diet, and his sexual activity. Knowledge encompassing desirable activities, competent performance, the commitment to enhance health in others, these are all learned from the example of peers or elders. These personal activities are shaped and conditioned by the culture in which the individual grows up, patterns of work and leisure, of celebration and sleep, of production and preparation of food and drink, of family relations and politics. Long-tested health patterns that fit a geographic area and a certain technical situation depend to a large extent on long-lasting political autonomy. They depend on the spread of responsibility for health habits and for the socio-biological environment. That is, they depend on the dynamic stability of a culture. The level of public health corresponds to the degree to which the means and responsibility for coping with illness are distributed among the total population. This ability to cope can be enhanced but never replaced by medical intervention or by the hygienic characteristics of the environment. That society which can reduce professional intervention to the minimum will provide the best conditions for health. The greater the potential for autonomous adaptation to self, to others, and to the environment, the less management of adaptation will be needed or tolerated. A world of optimal and widespread health is obviously a world of minimal and only occasional medical intervention. Healthy people are those who live in healthy homes on a healthy diet in an environment equally fit for birth, growth, work, healing, and dying. They are sustained by a culture that enhances the conscious acceptance of limits to population, of aging, of incomplete recovery and ever-imminent death. Healthy people need minimal bureaucratic interference to mate, give birth, share the human condition, and die. Man's consciously lived fragility, individuality, and relatedness make the experience of pain, of sickness, and of death an integral part of his life. The ability to cope with this trio autonomously is fundamental to his health. As he becomes dependent on the management of his intimacy, he renounces his autonomy and his health must decline. The true miracle of modern medicine is diabolical. It consists in making not only individuals but whole populations survive on inhumanly low levels of personal health. Medical nemesis is the negative feedback of a social organization that set out to improve and equalize the opportunity for each man to cope in autonomy and ended by destroying it. Acknowledgements. This article is dedicated to Ivan Illich and Thomas McKeown. Had their writings been taken more seriously, the world would have been spared the AIDS panic as well as other perversions. I would also like to thank Volker Gildemeister, Meditel, London, for translation and constructive criticism, and of course, my family, Hans Walter Weigand, and other friends too numerous to list, for all their support. End of article. AIDS, Death by Prescription. By Heinrich Kremer, Stefan Lanka and Alfred Hassig. Continuum, July/August 1996. Protease inhibitors and antiviral drugs with mitochondrial toxicity,
AIDS treatment with consecutive death. The advertising drums are beaten hard all over the world today. The same doctors are calling for obedient candidates for their experiments and holding out the same promise of a cure who have poisoned countless AIDS patients by administering the DNA blocker AZT for the past 10 years in an attempt to hunt down the phantom HIV virus. The same doctors are now trying to conjure up a substance from the test tube under the magic name of protease inhibitor and to market it as having a limitless cure potential, although nobody in fact knows what long-term reactions this molecule, which has never been tested on man, may cause in the living organism. The victims and perpetrators have only recently come to realize that AZT, also known as zidovudine or retrovir, has, in countless cases, brought about the inevitable and slow asphyxiation of the patient's body cells, which are in particular need of oxygen, and hence the equally inevitable death by poisoning of those persons who are stigmatized as HIV positive or diagnosed as suffering from AIDS and who trust their doctors. Despite that realization, new test candidates are already being sought who will be voluntarily prepared, through fear of death suggested by the medical profession, not only to swallow AZT in combination with allied toxic substances, but in addition to take an inhibitor which has an incalculable impact on cell metabolism. A guarantee of success is secured in advance, as with AZT, because any fatal secondary effects of the mixture are described as an outcome of the phantom HIV infection. These are the self-same laboratory doctors and clinical practitioners who for years abused the confidence of anxious AIDS patients with the assertion that AZT would reliably, and with total certainty, prevent the proliferation of their phantom HIV. In reality, the substance AZT is absorbed by a primary route through the DNA gamma polymerase into the energy center of all body cells, the mitochondria. Without the activity of the mitochondria as former bacteria, no body cell can produce the necessary energy from oxygen and make it available for the whole cell metabolism in the bound state in adenosine triphosphate, ADP. The doctors who prescribe AZT have, however, denied this established fact and wrongly diagnosed the fatal consequences of AZT medication as the sequels of AIDS following a prior HIV infection. For example, clinical manifestations such as the wasting syndrome, HIV encephalopathy, cardiomyopathy, atrophy of the skeletal muscular system and opportunistic infections of all kinds affecting the patients are declared to be tragic consequences of AIDS. AZT manifestly also damages the mitochondria of the same microbes, protozoa and fungi, which have become adjusted to the cellular metabolism of the body in the course of evolution without being normally pathogenic. In the case of serious damage to their energy production, they may, however, undergo mutation into aggressive pathogens and may, known as opportunistic infections. The true opportunists therefore are the AIDS doctors who prescribe AZT. They have sought to drive out devils with Beelzebub. And by doing so they demonstrate their ignorance of fundamental biological processes in the human organism. But the dogmatic AIDS doctors have invented new tricks. Although, despite all the assertion to the contrary, no scientist has ever demonstrably produced a genome of the imaginary HIV which would be capable of causing infection, they announce that they have traced minute fragments of the genetic material of HIV in RNA form and enriched these fragments. Now they claim to be able to determine the precise quantity of HIV in the individual patient's blood serum. LT remains a secret of the AIDS doctors to explain how they are able to identify the part as a whole, without ever having seen the whole. By the same token, researchers could conclude from the sight of a footprint on the banks of Loch Ness that the monster of the same name really does exist. But they go on to develop a destructive logic on the basis of such arbitrary definitions. As the doctors claim on the pars pro toto principle that they can quantitatively determine the active amount of the HIV virus in the individual stigmatized patient, they now prescribe appropriate quantities of AZT and similar toxic substances as a cocktail for the patient. A sufferer who is purported to have many fragments of the messenger substances of the genetic material of the HIV phantom in his or her blood serum, is described as an unfavorable case and receives the poison cocktail in correspondingly high doses. Sooner or later the patient will be unable to escape his or her predicted fate because of the fatal toxic effects of the medication, especially as, depending on the individual patient's reaction, the poison cocktails are varied and supplemented by protease inhibitors. The purported viral load hides nothing but the measurement of particular messenger substances, RNA, in the blood plasma of selected patients. Sequences which resemble those that are defined as HIV-specific are then demonstrated. But it must be realized that such messenger substances occur in thousands of different variations, reflecting perfectly normal biochemical processes in the body, thousands of which take place simultaneously and in coordinated fashion in the metabolic interplay. Fluctuations, i.e. the increased or reduced occurrence of the sequences, are perfectly normal in this complex interplay of thousands of simultaneous metabolic processes. In the case of persons with a heightened cellular metabolism, e.g. persons under cell-destructive medication, AZT, DDI etc., and those suffering from multiple infections, these molecules may sometimes occur with a higher degree of probability, precisely because of the metabolic acceleration. The isolated presentation of measurements of a particular kind of sequence, which remains in any case totally impossible to quantify, is therefore clinically irrelevant in the absence of comparison with other molecules of this kind. There are also no comparative values which would enable any significance whatever to be attributed to these relative measurements. Proteases are in fact protein enzymes which split protein molecules into the length required in each particular case by the metabolism. They are naturally rendered inactive within and outside the body cells by special inhibiting molecules until they are recovered by complex interactions between many different molecules. The body constantly produces such protease inhibitors, e.g. heparin, and the heparinoids. The HIV hunters now claim to have produced protease inhibitors in the test tube which will specifically only inhibit those proteases that are said to be responsible for the proliferation of the hypothetical HIV. They want to measure the success of these protease inhibitors by a quantified reduction of the arbitrarily defined viral load, see above, and the relative increase of the T helper cells. In other words, one fiction, virus blocking, is legitimated by another, virus cell quantification. The temporary increase of T helper cells is brought about by the partial displacement of cells of this type from the bone marrow and other compartments into the bloodstream through temporary inhibition of the catabolic metabolism, which predominates in HIV-positive patients. However, in reality it is to be feared that sooner or later the unphysiological intervention in the complex interplay of body cell growth factors through artificial protease inhibitors will disturb equally vital functions of the basic tissue and cells, together with their mitochondrial energy centers, as is already the case when AZT and allied nucleoside analogues are administered. However, as no animal model is available for preliminary clinical testing, the HIV-positive patients and AIDS sufferers who go in fear of death must put their life on the line. Every test subject should therefore be aware that treatment with cocktails of AZT and allied toxic substances plus protease inhibitors may be equivalent to joining a suicide squad with a time fuse. Finally, attention is drawn to the healthy organism where proteases and antiproteases are in equilibrium. Heparinoids at the cell surface are the normal antiproteases. An imbalance can be corrected by oral administration of heparinoids in the form of extracts of cartilage, chondroitin sulfate, and agar extracted from seaweeds. We suggest that anti-HIV positive individuals take advantage of this simple and cheap possibility to correct a possible deficiency of antiproteases. Dr. Heinrich Kremer MD was medical director of the Specialist Clinic for Juvenile and Young Adult Drug Offenders for 5 German counties, including Berlin, Bremen, and Hamburg. With the German virologist Dr. Stefan Lanka he initiated the Research Group for Investigative Medicine and Journalism, Reg. Med immunologist Professor Alfred Hassig, Professor Emeritus at the University of Bern, is a former director of the Swiss Red Cross Transfusion Service, and former president of the Board of Trustees of the International Society of Blood Transfusion. With colleagues he formed the Study Group for Nutrition and Immunity. End of article. HIV Pictures, What They Really Show. By Stefan Lanka. It has been long known that what AIDS researchers have presented as photos of HIV show normal cellular particles in use for export/import and other tasks. As those particles are designed, in contrast to viruses, for cellular use only, they are very unstable when removed from their context, and not able to be isolated and photographed in an isolated state. Genuine viruses are so stable that it is easy, in order to prove successful isolation, to photograph them directly as three-dimensional particles in the electron microscope, without prior chemical fixation. In contrast, the cellular transport and other particles are so unstable, excluding cell organelles like mitochondria, the energy-producing sites which are able to be isolated in a stable form, they can only be photographed in a chemically fixed state, in cells, tissues, or in supernatants. As these particles are not isolated and therefore are together with other materials, the chemically fixed and resin-embedded cells, tissues, or liquids, the mixed material has to be cut in very thin sections, ultra-thin sections, to be able to see anything. It's not possible in the electron microscope to look through thicker sections. Of course, existing viruses can be photographed in ultra-thin sections too, but, and this is the point, in their isolated form. All that have been shown to us HIV are ultra-thin sections of cellular particles, 1, 2 photo of very small particles in ultra-thin cell line section. The particles are claimed to be HIV, but are cellular and not viral particles. They are normally referred to as virus-like particles, microvesicles, and microsomes. The debris on the lower part of the photo indicates that the particles are not purified or isolated. These photos are always published without any evidence that the particles are of viral origin. Computer-enhanced photo of a cell surrounded by small particles. The blue/gray stained particles, which are claimed to be HIV particles attacking or, depending on the publication, leaving a white blot cell, are artifacts of the staining and/or fixation process and at best may be cellular particles entering or leaving the cell, in a well-known process called endo/or exocytosis. These kind of nice photographs, made by Leonard Nilsson, have been published without any evidence that the particles have a viral origin. HIV researchers believe the AIDS virus looks like this, like a bomb or water, blood, mine. This model is based on the detection of cellular particles in cell lines under very special conditions. Such particles never have been isolated or somehow else demonstrated to exist as a virus or be of viral origin. This is nothing but a model based on a collection of proteins of various size, no other characterization, which by antibody detection, known as HIV or AIDS test, have been chosen to be said to represent parts of HIV according to the ill-minded and false concept of retroviruses and how they should look like. Go here for some info on some recently published HIV photographs. Here you will find a paper by Lanka. End of article. Rethinking HIV. Collective Fallacy. By Stefan Lanka. Continuum September/October 1996. It is a dire example of how a distinguished scholar who has contributed much to the advancement of science, now impedes further progress by his stubborn adherence to a dogma of his own creation. If he did not feel himself obliged to repeat things that are untrue just because they were once said, they would have become quite different people. Johann Wolfgang von Goethe, Maximen und Reflexionen, Textile 586. Readers will be aware that there have been a number of claims for, and responses to, Continuum's offer of a reward for the missing virus. These have ranged from requests for clarification as to the type of proof required, through dismissive comments that the proof sought was an irrelevance, to an outright claim to the prize by Professor Peter Duesberg. Readers will recall that the point of the whole exercise was consistent with my article explaining that HIV does not actually exist, as opposed to the more frequently asked question whether HIV is responsible for AIDS or not. The distinguished Australians led by Dr. Eliopoulos Papadopoulos have already provided a detailed reply to the Duesberg claim, so I shall endeavour to explain how the erroneous concept of retroviruses brought about the present situation. Duesberg's enormous services to mankind are beyond dispute. It is he who for nearly 10 years now has steadfastly and at great personal cost, been the anchor of sanity and decency in a world driven mad by the simple-minded HIV theory. Whether HIV exists and whether it causes AIDS is largely academic. When did you last come across a normal heterosexual, someone who does not derive his living from perpetuating the panic, who takes the slightest notice personally of the official story? In practice, Duisberg's claim to our unqualified gratitude has been his long-standing and unwavering opposition to AZT, and its analogues, whose use always ends in death. That said, it is unfortunately also true that Duisberg is himself the victim of another collective fallacy, the Denkollective of Ludwig Fleck, which he himself helped to formulate and which he is now apparently locked into. Retroviruses were postulated as being that species of microorganism which caused reverse transcription to occur, which, as a working hypothesis at the time in the early 1970s, was entirely reasonable. The mistake was to elevate the hypothesis to a dogma. Early gene detection techniques lent some credence to the existence of an entity that could be transmitted from one cell to another, which was unfortunate, because this, too, turned out to be wrong. Errors of this kind occur whenever technology makes available for general use a new experimental procedure, which propels a whole army of researchers into mass-producing experimental data, heedless of what the biological significance, if any, of their work might be. Even worse, is the habit of making countless ad hoc adjustments to the original theory, which completely distort the original hypothesis. Correct science demands that there should be a radical rethink when this happens. If there isn't, as in this case, a fundamentally flawed concept goes haywire, ending in disaster. Duisberg went along with mainstream AIDS researchers, limiting his objections to the relatively minor aspect of whether HIV could cause AIDS or not, whereas he really ought to have smelt a rat regarding the whole concept of retroviruses, given his earlier, courageous stance in admitting the mistaken role of retroviruses in causing cancer long before anyone else did, even though he was deeply enmeshed for a long time in that fallacy, too. In my view, he could reasonably have been expected to satisfy himself that there was in fact no such an entity as a retrovirus at all. Instead, he has allowed himself to be bedazzled by the technical feat of retrovirologists who manage to reproduce in a consistent manner certain phenomena peculiar to particular biological constituents of cells. In so doing, he allowed himself to be misled into believing that this was due to a virus. It is a complete non sequitur. This lack of intellectual rigor has, in a contemporary metaphor, debased molecular biology to a virtual science, leading to the deplorable state of having a disease, AIDS, with a virtual definition, due to a virtual pathogen, HIV. Unfortunately for humanity, AIDS is not unique in this regard, but represents merely the tip of a much larger iceberg. To the astute observer, it should have been apparent as early as 1973 that the working hypothesis of ascribing the experimentally observed phenomenon of reverse transcription to retroviruses had become untenable. When it became known that reverse transcription was anything but rare, by 1980 at the latest, the hypothesis should have been abandoned by everyone. Indeed, the extraordinarily artificial and circumscribed conditions under which reverse transcription could be induced in the laboratory should have alerted everyone to the extreme improbability of such exclusively laboratory conditions having any bearing whatsoever on naturally occurring phenomena. All the more so, as no retrovirus was ever shown to exist, for example, by being able to isolate and characterize it, and to demonstrate its transmissibility. These failures, obviously not for want of trying, should have sufficed to kill off the whole concept. It may be hard to believe that all maps purporting to represent a whole retrovirus, including HIV, are always compilations, many bits and pieces cobbled together by their authors to the best of their beliefs. They are collages. No complete retrovirus nor its RNA in its entirety has ever been proved to exist either in vivo or in vitro. A further difficulty for the hypothesis was that it had never proved possible to show that the experimental observations attributed to retroviruses were exogenous to the cells used in experiments, i.e. that they came from outside of the cell. Indeed, all the evidence pointed to the opposite, i.e. that they were endogenous, inherent, to the cells themselves. Part of the evidence was that the so-called retroviral activity could only ever be induced experimentally in one type of cell, whereas HIV is supposed to infect many different types of cell in the body. The two contentions are clearly incompatible. The whole theory is rendered even more implausible when it is remembered that retroviral concentrations are always extremely low, which is why a huge excess of cellular material from patients is needed to be able to demonstrate any replicating virus at all. This, incidentally, is also the basis for the claim that HIV has only a low rate of infectivity. A much more rational explanation is that there is no virus there at all. History furnishes an unhappy precedent for this form of research. At the turn of the century, experiments were conducted using highly inbred laboratory animals. Under strictly circumscribed conditions, these displayed higher disease susceptibilities than animals which were not inbred. The phrase highly inbred was forgotten about, and generalizations about viral infectivity were made which turned out to be wrong, from which medicine has not recovered to this day. In like manner, experiments are nowadays performed with cell cultures instead of whole animals, for the very good reason that they greatly speed up experiments. The disadvantage is that this limits experimentation to just one of a few cell lines, which are always cancerous, because only they will grow continuously in the laboratory. History is repeating itself. Generalizations are made about the behavior of normal cells on the basis of results obtained from highly abnormal cells. Such cells can incorporate extraneous pieces of DNA into their own DNA, when added to growth medium, as normal cells can, too, only more slowly. Cells, which have incorporated such DNA, will obviously manifest characteristics for which the DNA coded, making it appear that a virus had been at work, when nothing of the sort had happened. It is easy to see, therefore, how the bizarre notion of infectious DNA arose, and to conclude, wrongly, that a virus, in the conventional use of the word, is involved. The whole argument collapses, however, when it can be shown that non-viral DNA can be made to do this too, both in vivo and in vitro. If that DNA happens to be DNA arbitrarily defined to be HIV DNA, or part thereof, then clearly the cell, which has incorporated the DNA, will behave as if it had been infected by HIV. Yet this is the basis of Duesberg's claim. In his retrovirological zeal he does not seem to appreciate that infectious DNA is a contradiction in terms. For what else is a virus but that? Folded up DNA wrapped in a protein coat so that the DNA can be transmitted from one cell to another, is what is normally called a virus. A loose strand of DNA could not do this by itself, it would be exposed to enzymic degradation, it would become entangled with other components. How would it identify its target? How would it get there? How would it enter it without a specific mechanism? A man of Duesberg's abilities should need no prompting that replicating equals cloning something in a test tube and then detecting that something equals molecularly cloned DNA, in a place where you have previously put it, is a circular argument, and therefore no argument at all. But then, tautologies are an indispensable part of all retrovirology, as I was at pains to point out when explaining the fallacy inherent in HIV antibody tests. Conclusion. The rules demonstrating the existence of HIV, and retroviruses in general, were never adhered to by those who devised them, nor were they ever validated. It is now easier to understand why people felt it necessary to inquire of this magazine what the rather self-evident term isolation meant. Suitable synonyms might be pure and/or free of contaminants. They clearly had a slight worry at the back of their minds all along that the term was used in retrovirology rather as in Alice in Wonderland it means what I say it means. Until AIDS was invented, retrovirologists were a minority sect who were happy to accept each other's flights of fancy without being too critical. They could fiddle around to their heart's content, safe in the knowledge that retroviruses were the least dangerous of all viruses. Well-meaning and credulous colleagues, as well as aspiring virologists, journalists and, through them, laymen were mesmerized by incomprehensible jargon into believing that the mass of data on HIV and retroviruses somehow meant something. Each property relating to HIV, and retroviruses generally, can be shown to pertain to the cells used in the CO cultivation experiments. At no time have there ever been any credible grounds for thinking that these properties and components had anything to do with viruses in general, nor with HIV in particular. No particle of HIV has ever been obtained pure, free of contaminants, nor has a complete piece of HIV RNA, or the transcribed DNA, ever been proved to exist. Asterisk. End of article. Lanka replies to Duesberg, 2. The latest reaction of Peter Duesberg, 1, to the ascertainment that HIV does not exist, 2, and to the thorough line of argument that all claims about and characteristics ascribed to HIV do not withstand specific scientific examination, 3, raises questions. Why does he so vehemently defend something for which there are not only no proofs but also no necessity, and which has pushed millions of people into fear of a retroviral plague transmitted through sex and blood? Why do HIV virologists never subject their viruses to the same generally accepted standard techniques of molecular biology as all other virologists and biologists do? In his latest monograph of May 11, 1996, Peter Duesberg introduces a more untenable claim than ever, which neither he nor anybody else can substantiate, to suggest again that there is a genetic entity HLV. He equates cloning, a standard technique of multiplying a given genetic sequence, with virus isolation and the existence of HIV. After the question of the existence of HIV was first posed and people began to rethink HIV and to understand that with the available, exact identification techniques of genetics, biochemistry, and virology not a single aspect of the existence of HIV has been proved, apparently there was a desire quickly to postulate new criteria for its existence. That this new line of argument is put by Peter Duesberg, known for his critique of the idea of an infectious AIDS yet otherwise one of the godfathers of retrovirology, is revealing. But the quicksand beneath Duesberg's construct is visible. When instead of referring to the established criteria of structural identification, he now postulates functional criteria, cloning, multiplying, is isolation, though the thing to be cloned has never been identified as part of HIV. No structural criteria with which one can exactly identify genuine biological entities are to be used in the case of HIV, no analysis of the form and size of an isolated virus, the kind and composition of its proteins, e.g. if one wants to use the proteins in an approved antibody test, its genetic substance, e.g. if one wants to carry out the test tube experiments which Duesberg cites or do viral load measurements. We are encouraged arbitrarily to believe in only the repetition of processes, which ad hoc were ascribed to be viral attributes. When he claims there are at least 19 full-length HIV genomes, that 19 molecules of the complete genetic substance of HIV exist in this world, though this has not been shown or claimed in a single scientific publication. For the purpose of secure identification of a virus, the right means, the means of structural isolation, have to be applied before one carries out functional examinations with parts of the virus. For a clear understanding of this important argument, the main terms are again briefly explained here. A virus is an acellular form of organism, being no more and no less than a piece of genetic substance, according to a given species of virus, always of the same length, and a covering surrounding the genetic substance, composed mainly of proteins, according to a given species of virus, always of the same form and size. Viruses are stable because they have to leave cells or even the organism in order to infect other cells or organisms anew. Using centrifugation techniques, it is no problem to separate viruses from all contaminating components, and in doing so to isolate them, then photograph them, then represent their proteins and genetic substance in a direct way. In the case of HLV, this has not been done up to today, for HIV as a whole, or therefore for any of its components, its proteins or genetic substance. 2, 3. The scientific conclusion is that the existence of HIV has so far not been proved. The logical explanation, given that all characteristics ascribed to HIV are well-known cellular entities and characteristics, is that HIV never was, and the claim of the existence of HIV is not sustainable. The idea which led to the claim that HIV exists is based on a decisive false assumption. From 1970 on, some scientists and much of the public were led to believe that since a certain biochemical function, reverse transcription with its then unfamiliar mode of action, did not fit the dominant world picture of genetics, it would be explained only through the claim of the existence of a new class of viruses, the retroviruses. The shock of reverse transcription was that it is possible to make genetic substance out of messenger substance, which until then was believed to be impossible. However, that the detection of reverse transcription is not, as some research directions still assume, a sign of certain death, e.g. HIV equals AIDS equals death, Gallo, Ho, and colleagues, or a reference to the most harmless viruses in the world, Duisberg, was proved when it was shown that reverse transcription reflects a repair mechanism of damage in cellular genetic material, in one revealing experiment, the chromosomes of yeast. 5. So tragically, in 1970 the detection of a healing process gave birth to the idea of a new class of viruses and eventually HLV, because astonishingly researchers were not willing to rethink their models or listen to what nature has to tell them. The stubbornly held notion that reverse transcription was inevitably retroviral was first employed in the war against cancer as cell-multiplying viruses, then as the opposite, in the war against, medically induced, AIDS as cell-killing viruses. It is of the greatest importance in this context that HIV researchers, when trying to detect the activity of reverse transcription, which is always the first step in the attempt to identify retroviral structures and characteristics, instead of using the natural genetic messenger material, the RNA genome of the virus which should be there if the viruses existed, always use, without any explanation why, synthetic messenger material templates. 6. Above all, it is known that those templates are not specific for the process of reverse transcription, that they are efficiently recognized and transcribed by the normal, common, cellular genetic material producing enzymes as well. 3. The whole idea of HIV would collapse if it was possible to bring this fact to public attention. It should be clarified, it is very normal that genetic material DNA, natural or artificially multiplied, when put onto cells is able to enter those cells, may integrate itself into the cells' chromosomes and eventually may be activated to produce its proteins. The idea of vaccination with naked DNA, to which I strongly object for various reasons, is based on these known mechanisms. To add a DNA clone to cells and later to prove its presence and probable activity is nowadays a standard experiment in lectures on biology, but in no way a proof for the existence of HIV. So one can only guess why molecular biologist Peter Duisberg refers to such a standard experiment as proof of the existence of HLV. As the group around Eleni Eliopoulos et al. Has shown, 3. Neither he nor anybody else has shown that the genetic pieces of HIV used in the transfection experiments he cites, 9, were isolated out of a virus. Only if researchers were able to multiply from cells exactly that genetic material which previously had been isolated from a virus, only then the claim of virus detection would be valid. Virus isolation logically always goes first. Or may anybody postulate new viruses, sprinkling his or her genetic material onto cells, detecting this material in the cells and claiming a new virus? A repeated artifact remains an artifact. To call such redetected DNA infectious DNA is conspicuously misleading. When Peter Duisberg refers explicitly to a publication in which we read, tested blood cells of 409 antibody positives including 144 AIDS patients and 265 healthy people. In addition 131 antibody negatives were tested. HLV-specific DNA subsets were found in 403 of the 409 antibody positives, but none of the antibody-negative people. 10. While these claims and statements are not at all substantiated in the further reading, 3. It becomes obvious that he simply cannot have correctly read this publication, which clearly touches clinical aspects, and that he thereby risks grossly violating his scientific ethics. And when those ethical gentlemen, Duisberg's colleagues, two of the world's leading retrovirologists Robert Gallo and Robin Weiss, are invoked, without doubt about their claims, either for having re-isolated only HIV from Montanier's virus stock without recognizing any further contamination. Peter Duisberg's pseudo-rationale needs an explicit comment. If one looked for other retroviruses, in Montanier's stock, they would be found in great numbers these days. The human genome carries thousands of such genes that can be traced back to the action of reverse transcription. 3. And were named retroviral elements by retrovirologists in an ad hoc decision. 2. In Robert Gallo's humiliated hunt for a credible new retrovirus, he was not taking care over or notice of grossly contaminating viruses but instead mixed up together, it actually happened, the materials of 10 patients, in order to be able to create HLV. 2. Gallo himself wrote in the very important statement of the September 27, 1983, after the decisive conference in Cold Spring Harbor, the virus described by Montagnier I have never seen sick, and I guess that he has a mixture out of two. On the other side, some of his data are interesting but not at all conclusive. When Duesberg urges, as I pointed out in my missing virus reward claim in the July/August continuum, infectious HLV DNA has been isolated from infected cells several times by molecular cloning, he himself is honest enough to claim that this infectious HIV DNA has been isolated not out of LDS patients but from special infected cells. He conceals that those cells underwent a very particular treatment, and had DNA added before. 2-3. He cites only literature in which ex cathedra the same claim as his is made without a single reference that the importuned infectious HIV DNA was detected in or isolated from a virus. 3. Predictably, 3 references, Fisher et al., Barnett et al. And Levy et al., which Duesberg cites, to support his claims reveal that phenomenon typical of AIDS research. In the headline and the abstract of the publication things are exactly named and specific claims are made that are wholly unsubstantiated in the further text. One has only to read the small print of the technical comments, examples are commented on in Ref 3, NB pages 16 to 18, to see and understand the misconceptions behind them. When Duesberg lends his reputation and charismatic authority to such duplicitous science with its fatal consequences, without any reflection of the detailed critique, 3, and especially without any analysis in his field of expertise, it is very precarious and alarming. The explanation for his contrived insistence with highly technical and quasi-exact vocabulary that contrary to the complete case of Papadopoulos et al., 3, there is somewhere out there a HIV, detectable only through cloning because cloning, is in fact the most rigorous isolation science has to offer for retroviruses, may be that though a genuine retrovirus HIV has never identified, retrovirologist Duesberg can't or doesn't want to admit it, for reasons that may not be obscure. But it is increasingly beyond indulgence that Duesberg piles up claims of 19 complete genomes, complete genetic sequences, of HIV which it has been possible to artificially multiply in the form of clones, then, to build up theories of probability bereft of significance gives the impression that they all have the same length when no scientist before him has ever claimed this, ever seen such things or of course ever published such claims. LN the only reference he could have meant, 4, it's enough to read the title Recovery of Virtually Full-Length HIV-1 Provirus of Diverse Subtypes from Primary Virus Cultures Using the Polymerase Chain Reaction, to understand that Duesberg wildly appropriates references that seem suitable for his purpose without actually knowing them. The cited method, the polymerase chain reaction, PCR, is not able to construct something like a viral genome. 12. It certainly has to be clarified that concocted and size-selected genetic molecules like HLV clones can never represent an almost theoretical isolation somehow on account of their lengths being in a proportion of 1 to 100,000 against the length of the full human genetic material. Such clones result from the process of concocting smaller molecules, produced in a test tube using the cellular genetics and cellular enzymes, and then, to present a whole sequence, uniting the smaller sequences in theory, on paper, or in a computer, following a blueprint from the arbitrary rules of retrovirology. LT beggars amazement how one may state ex cathedra that, the high standards of virus isolation, may be relevant for crystallographers or chemists, but are not relevant for functional isolation, when it is those unspecified HIV proteins created in cultured cells when HIV researchers tried to induce reverse transcriptase activity that are used in the AIDS test, which is an instrument of sentence over death and life. What by God is then relevant in this kind of science? Not relevant for functional isolation. What for heaven's sake is meant by functional? That the LDS test works when one believes in its function? And it is incomprehensible how AIDS expert Professor Duesberg continues to discuss AIDS as if there were an autonomous clinical picture which one may call LDS. Every concerned person knows by now that AIDS has a different meaning on every continent, with its different causes. AIDS is an inadmissible artificial diagnosis, it has been legitimized through the introduction of HIV as a constructed cause. The AIDS myth with its exploitation of the fear of an alleged deadly sex plague has been blindly launched into the realm of seemingly scientifically based biological fact through the pseudo-rational HIV detection technique. But the two terms according to the rules of construction depend on each other. The clarification of the question whether HIV exists, with the most secure method of identification available, and this would be only the isolation of complete high viruses, is a sine qua non for dismantling the mass delusional trance called AIDS. Only then may the task of research with the needed temperance and precision, into the causality of the concrete disease complexes behind AIDS, be addressed. 14. The danger that the AIDS critical movement splits or temporarily weakens over the question whether HIV exists is therefore secondary, and in any case subordinate to, the right to life of every person. The common matter is to secure the human rights of every stigmatized person, not a personal cult. The human rights do include free, and in this case essential, access to information. Censorship, and worse, misleading, for political or personal reasons, be it ignorance or the will not to know, is endangering human lives. LT is urgent. The one who comes too late is punished by life. General political knowledge. Those who too late or never receive essential knowledge may die in the throes of an AIDS diagnosis or commit suicide. Sad, bad wisdom amongst HIV and PWAs. That HIV has never been identified as secure biological matter is of the greatest importance and must immediately be told to every stigmatized person. No HIV, no false diagnosis AIDS, no death sentence, no false treatment, no unnecessary suffering, no needless dying, but new chances for people who for complex reasons got seriously ill, amongst them being labeled as AIDS cases and HIV positives et al., and then falling victim to medical short-sightedness based on laboratory technical constructs. There is no high virus. Stupid? Asterisk. Stefan Lanka. Source: Continuum February/March 1997. End of article. 15 years of AIDS. The continuous failure in the prevention and treatment of AIDS is rooted in the misinterpretation of an inflammatory autoimmune process as a lethal, viral venereal disease. By A. Hassig, H. Creamer, S. Lanka, W. X. Liang, K. Stampley. May 1998. Summary: the question of the specificity of the anti-HIV antibody test has to be re-evaluated as it was shown that the viral enrichment obtained from CO cultivations of patients' lymphocytes with fetal cord blood by Barcinicea etal and leukemia cells by Gallo etal, exclusively consisted of proteins of the cell types used in the cell culture. This precludes a clear separation of presumed retroviral and cellular proteins or extracellular matrix proteins. In this context, it was shown that the anti-HIV antibody test detects autoimmune antibodies directed against cytoskeletal proteins, e.g. the liver cells. Strongly augmented anti-actin autoantibodies is considered close to pathognomonic for chronically active hepatitis. The original assumption that reverse transcription from RNA to DNA is evidence for the existence of retroviruses was wrong. In fact, reverse transcription is a vital mechanism for the maintenance of the genome. The decrease in numbers of circulating CD4 lymphocytes can be explained by a stress-induced hypercortisolism. Up to date, direct HIV-mediated destruction of CD4 lymphocytes could not be proved. The same is true for measuring of the viral load. Shortcomings of the applied method to quantify the viral load do not permit definitive conclusions. Possibly, it may be taken as an expression of a stress-induced weakening of the cellular immune reactions, in the course of which the nucleoside fragments resulting from the current cell turnover are inadequately eliminated. Furthermore, the treatment of patients with nucleoside analogues has a toxic effect on both the genome of the cell nucleus and the mitochondria. The latter, therefore, may produce insufficient amounts of ADP, causing organ failure and, eventually, death. The synthetic protease inhibitors used these days are associated with serious side effects. Therefore, it seems worthwhile, in these patients, to bring back the catabolic situation due to whole body inflammation to homeostasis by administering anabolic phytopolyphenolic compounds. AIDS is the abbreviation for Acquired Immunodeficiency Syndrome. AIDS, as a term for an illness, originated in the search by the American Centers of Disease Control for sick homosexual men, also suffering from Kaposi's sarcoma, KS, and/or pneumocystis carinii pneumonia, PCP. In 1983, Harrison-Chirimuuta et al. Reported on a T lymphotropic retrovirus which they allegedly isolated from an enlarged lymph node of a homosexual patient. 1. In 1984, Gallo et al. Reported the alleged isolation of an identical retrovirus from CD4 lymph cells from homosexual patients, clinically diagnosed as suffering from AIDS. 2. Harrison-Chirimuuta et al. Cultivated these patients' lymph cells in question with fetal cord blood, and Gallo et al. Cultivated theirs with leukemia cells. Initially, these laboratory methods must raise doubts as to whether the isolation of a new human retrovirus is evident just by these data. Gallo et al. Stated that their allegedly isolated retrovirus caused the destruction of CD4 lymphocytes in those patients, whose heterogenic illness was taken as an after-effect of the CD4 cell destruction and so subsumed as AIDS. Besides, Gallo et al. Announced that in due time a vaccine would be available for the formation of antibodies against the discovered virus. 2. Today, 15 years later, the question still remains open, whether high retroviruses actually do exist or whether the postulated retroviral HIV antigens as well as the postulated HIV reverse transcription are a matter of human protein molecules derived from cells in CO-cultured cell cultures used by both Harrison-Chirimuuta et al. And Gallo et al. The most extensive investigations in this regard are owed to Eleni Papadopoulos-Eliopoulos and her group in Perth, Australia. In 1993 they published a review concluding that there is no evidence for the existence of high viruses. 3. In 1994 Lanka demonstrated that all retroviruses, including HIV, are biologically inexistent and their phenomenology is based on laboratory artifacts. 4-6. Eleni Papadopoulos confirmed Lanka's interpretation in a recent comprehensive review. 7. These fundamental counterstatements to the current HIV/AIDS theory have been strongly supported in the last few years. Upon investigations in order to develop a vaccine against HIV, it became apparent that the enrichment of presumed HIV-1 preparations, considered as pure, consist of proteins of the cell types used in the cell cultures, which resist a clean separation into presumed retroviral and cellular proteins, i.e. extracellular matrix proteins. Above all, these cell proteins also occur in the inside of extracellular particles, which have been misread as it seems as so-called high virions by the retrovirologists. 8-10. These findings were to be expected as Gallo et al., when developing the AIDS test, did not investigate the presence of cells' own proteins in the protein mixture, released during the CO cultivation of patients' lymphocytes and leukemia cells. Upon developing the ELISA and Western blot tests, it should have been imperative to consider proteins released from stimulated leukemia cells, not being mixed with patients' lymphocytes, and to differentiate these from the ones released only after addition of patients' lymphocytes. In view of this, it seems to be mandatory to re-evaluate the question of the specificity of the anti-HIV antibody test. What is the laboratory finding anti-HIV positive based on? In a series of preceding reports we have discussed this question in detail, 11-14. We came to the conclusion, the laboratory finding anti-HIV positive is primarily the expression of an autoimmune activation of the immune system linked to a persistent catabolic state of metabolism. In view of the fact that the diseases grouped under the term AIDS are limited to risk groups such as homosexuals, drug addicts, and recipients of blood products contaminated with parenterally transmitted hepatitis inductors, the question is raised, whether the anti-HIV test determines autoantibodies directed against cell envelope structures with a specificity to the body's own proteins of the host cells. It has been known for over 20 years that chronically active hepatitis, currently hepatitis B, hepatitis C, and autoimmune hepatitis without evident antiviral antibodies, react by the formation of autoimmune antibodies directed against cytoskeletal proteins of the liver cells. Thus, the raised anti-actin autoantibodies are pathognomonic for chronically active hepatitis. 15.
Johnson et al., in 1965, were the first to report on anti-actin autoantibodies, 16. They described autoantibodies directed against smooth muscle cells and showed that this had to be considered as characteristic indication of lipoid hepatitis. In 1973, Gabuni et al. Demonstrated that autoantibodies directed against smooth muscle cells react with actin-containing microfilaments, 17. Further investigations indicate that autoantibodies with anti-actin specificity are to be classified within the big group of autoantibodies against filamentous proteins of smooth muscle fibers. 3 to 18% of healthy individuals present low-titer autoantibodies against cytoskeletal proteins, 18. High-titer anti-actin autoantibodies, on the other hand, are only found in patients suffering from chronically active hepatitis and/or biliary cirrhosis, 19. In 1994, Burmese et al. Showed that both sera from patients with lupus erythematosus and from mice suffering from the same illness react with glycoprotein 120 and peptides of the postulated HIV-1 envelope, 20. They further proved that control sera of healthy individuals and patients with other autoimmune diseases contain small amounts of the same autoantibodies. Last but not least, they showed that autoantibodies reacting with glycoprotein 120 do not possess antinuclear specificity. They refrained from investigating a specificity against cytoskeletal proteins of these autoantibodies. Evidence is given that the anti-HIV test does not indicate antibody formation against the postulated retroviruses as, during the last decade in Germany, not a single seroconversion has been observed in imprisoned drug addicts. All seropositive drug addicts acquired their anti-HIV positivity before their imprisonment. In opposition to this, seroconversion by hepatitis B inductors was recorded in intravenous drug addicts, 21 to 23. The same was also observed in hemophiliacs, i.e., despite continuous substitution with hepatitis-contaminated blood products, approximate a third of these individuals never become anti-HIV positive. This is characteristic for the individual response to autoimmune reactions against cytoskeletal proteins in the host cells, in which Gerard and Sen, Cal observed a polyreactivity, 24. The individual autoimmune reactivity either appears at first contact or fails to appear even at multiple contacts. We conclude that a positive anti-HIV test does not indicate an antibody formation against retroviral HIV antigens. Low-titer anti-HIV antibodies are common even in healthy individuals. High-titer anti-HIV antibodies are pathognomonic in chronically active hepatitis. The anti-HIV test does not answer the question whether anti-HIV antibodies occur or not. The test differentiates between plenty equals positive and few equals negative. Rethinking as to reverse transcription. The error, taking proteins resulting from HIV isolation for retroviral proteins, dates back to 1970. The paradigm, that DNA codes information and programs relating to all physiological and phenomenological aspects of all organisms, resulted in the postulation of the irreversibility of the genetic flow of information for the synthesis of proteins, from DNA via messenger substance, RNA, to proteins. This was the crucial genetic dogma. 25. Despite the proven reversibility in 1970, that from RNA DNA can remerge, this fact was postulated as an exception that proves the rule by stating the existence of retroviruses, qualified for this reversibility, which, at this time, were only considered as two more viruses, 26, 27. With the discovery of this enzymatic activity in all living cells, it soon became clear that the evidence of the function of reverse transcription from DNA into RNA was not a proof for the existence of retroviruses, because the genome of all eukaryotic cells is clearly marked by this activity, 28, 29. Retrospectively, it seems rather astonishing that in 1983 Montagnier and in 1984 Gallo still postulated a new retrovirus despite the fact that a new viral entity had never been isolated or described, according to the standard regulations in virology. As a matter of fact, the enzyme reverse transcriptase from OHIVO has never been isolated or described, but only inferred from functions to its existence, when new formation from DNA into RNA was proven by laboratory techniques.
Since 1985 it has been known that the reverse transcription plays a decisive role in the maintenance of the structure of the genome, by repairing chromosome fractures and, especially, by limiting the loss of chromosomal end components, the telomeres occurring at cell replication, 30 to 33. The respective enzymes for this kind of reverse transcription, the telomerases, dispose of a type-specific RNA matrix for the formation of the repeating telomere units. Somatic human cells, that do not belong to the reproductive path, cannot adapt to the shrinking of their telomeres when replicating and so cease the replication at a certain degree of depletion. Up to date, the influence of nucleoside analogous on the action of telomeres at replication has obviously not been investigated. We could trace only two publications, in 1996, which describe an in vitro investigation of nucleoside analogues inhibiting the telomerase activity. In our view, the knowledge, already gained in 1980, on the vital physiological function of reverse transcription, should have led to a rethinking as to the establishing of nucleoside analogues as pharmacological inhibitors of the reverse transcription of the postulated high viruses and, according to knowledge of that time on the physiological function of the reverse transcription, it should have been rejected. 34-35. What is the decrease of CD4 lymphocytes in AIDS based on? The decrease of the circulating CD4 lymphocytes in the bloodstream during progressive immune deficiency in AIDS has generally been explained by the progressive destruction caused by high viruses. 36. 4 years ago Carbonari et al. Showed in an in vitro investigation that the apoptosis of the lymphocytes in AIDS patients is mainly related to CD8 T cells and CD19 B cells. 37. Finkel et al. Then pointed out that apoptosis concerns mainly bystander cells and spares supposedly infected cells from so-called HIV and SIV lymph nodes. 38. These reports remind us of Fossey's classical publications of the 70s in which he and his working group clearly demonstrated that, in persisting hypercortisolism, an increasing number of CD4 cells leave the bloodstream and can thus activate B cells in the marrow. 39-44. The migrated CD4 cells return to the bloodstream upon dropping to normal values of the cortisol level. At the beginning of 1995 Wei and Ho et al. Published a report in which they declared that the extremely fast multiplication of Hi-1 viruses produces a raised turnover of CD4 lymphocytes. 45, 46. Towards the end of 1996 Wolders et al. Showed that the telomere length of CD4 lymphocytes in anti-HIV positive individuals remains normal, whereas the one of CD8 cells decreases. 47. During the latest International Congress of Leading HIV Scientists the long-term criticism of the HIV/AIDS theory has been confirmed. Despite intense and precise investigations there was no proof of a pathophysiological mechanism explaining the different reaction of CD4 and CD8 lymphocytes to the postulated retrovirus HIV. 48. It was literally stated, the riddle of CD4 cell loss remains unresolved. Paul Johnson of the Harvard Medical School in Boston voiced in a disillusioned way the helplessness of the conventional AIDS scientists. We are still very confused about the mechanism that leads to CD4 depletion, but at least now we are confused at a higher level of understanding. In other words, Fossey's pioneering work, based in the 70s, on experimental traumatology fell into oblivion. Calvano clearly documented in a review published in 1986 that the selective depletion of CD4 lymphocytes is induced by neuroendocrine mechanisms in traumatic conditions such as injuries and burns, whereas its proportions depend on the degree of hypercortisolism. 49. It remains enigmatic why Fossey, after having joined the AIDS research, never again mentioned his own reports. What does the viral load measure? Immediately, after the publication of Wei's and Ho's reports in January 1995, 45-46, in which they put forward the hypothesis that high viruses multiply at raving speed destroying a similar number of CD4 helper cells, quantitative tests based on the genetic multiplication method PCR were introduced and so a large number of HIV in the bloodstream was postulated. It has been well known among HIV scientists that the so-called viral load, i.e. the measurement of the viral load, is no evidence of the entire virus genome or intact viruses. 50.
The viral load measures short components of the messenger substance RNA, attributed to the high viruses. Because the high virus genome per SE never could be described, it is impossible to designate these RNA fragments as viral. Going through the records of the presumed characterization of HIV, it can be inferred that all components, proteins and genetic substance, attributed to HIV, are of pure cellular origin, 3 to 7. Therefore, the results of the viral load can only have an indirect significance, such as the measurement of an increase or decrease of cellular RNA, as it can be observed as increasing in catabolic conditions of cell disintegration and decreasing in anabolic conditions. However, these results cannot be considered as clinically relevant as, besides the technical inadequacy, control investigations with both non-positive defined healthy and ill individuals have never been published. The polymerase chain reaction, PCR, is a technique for a manifold multiplication of short DNA fragments, developed by Nobel Prize winner for chemistry, Harrison-Chirimuuta. Upon measuring the viral load, the RNA fragments in the bloodstream first have to be converted into DNA and then multiplied as such. The single, developing technical steps of this method are prone to failure. The slightest impurity, drugs, such as heparin and other substances, interfere with a reproducible functioning of the PCR method, especially with quantification. 53. Harrison-Chirimuuta, the inventor of this method, does not miss any occasion to criticize the application of his technology in the context of AIDS. 52. Furthermore, it is concealed that it does not make sense, either practically nor theoretically, to initially multiply manifold fragments of genetic structure and then to postulate their manifold presence. In case they were actually present in the blood samples, it would cause no problem to prove their existence by simple, quick, and cheap standard methods. 51, and, if de facto, viruses did exist in the bloodstream, scientists certainly would have been successful in making them visible. Hence, up to date, no scientist claims this achievement, a fact which has been confirmed under coercive evidence by the German Health Ministry in 1996. After the report of a produced positivity in the viral load during a vaccination test with proteins, 54, of a previously negative defined test person, it is now frankly admitted that repeated wrong positive results in the viral load are quite a well-known phenomenon. 55. Impairment of energy formation in mitochondria by nucleoside analogues such as AZT, azidothymidine, zidovudine. AIDS patients quite often demonstrate a weakening of their skeletal muscles. Up to 1990, this was considered a high virus-caused impairment of muscles. In 1990, Dalaka's et al. Demonstrated that this kind of muscle disease is due to an administration of AZT, weakening the mitochondria within muscle cells. With the excessive release of free radicals, the mitochondria are affected in their function of forming ADP as key substance in metabolic energy. 56. Hayakawa et al., in 1991, demonstrated important changes in the mitochondrial DNA, mtDNA, in the liver of mice after the administration of AZT. The final sentence of this paper reads, however, for AIDS patients it is urgently necessary to develop a remedy substituting this toxic substance AZT. 57. These results were confirmed by histochemical methods in the same year by Chariot and Gherardi. 58. The toxicity of nucleoside analogues in the treatment of viral diseases was thoroughly dealt with in the following years, and it was proven that the toxic effect causes multi-organic impairments in heart muscle, brain, and kidney, as well as in liver and pancreas. 59. Further, it was shown that successor drugs of AZT such as DDL and DDC cause the same mitochondrial impairment. 60. Since 1991, it would have been mandatory that not only the pharmaceutical industry but also the registration authorities seriously consider these impairments caused by long-term administration of nucleoside analogues and provide proof of the incoherence of AIDS patients' death with this drug treatment. In general, this obligation has been avoided and now they face upcoming connected liability questions. HIV proteases inhibitors, a new therapeutic principle in the prevention and treatment of AIDS. According to the HIV model, long precursor molecules of proteins along the multiplication process have to be cut at certain interfaces in order to create functional HIV proteins upon which, ultimately,
new high viruses form. Synthetically produced short protein molecules, reproduced after the interface to be cut from the precursor protein but which cannot be cut should, according to the model again, inhibit the natural activity of the HIV protease and thus prevent the formation of new high viruses. As a matter of fact, the HIV protease has not been isolated, but has been reconstructed by genetic engineering, upon which it was observed that this enzyme is very similar to the human digestive enzyme, pepsin of the class of the aspartate proteases. The problem of the model is that the one and the very same HIV protease would have to be cut at completely different interfaces in order to form functional proteins and, ultimately, HIV. Practically, this is not conceivable and has been explained as, enzymes do not have a high sequence specificity, although it has been postulated that, a therapeutically applicable inhibitor has to be specific, and should not inhibit human enzymes of this class of substance. 61. Considering these explanations of the head of the chemical department of the scientific laboratories at Bayer's, it becomes obvious that, theoretically, it is not possible to exactly target the postulated HIV protease. Further, it is impossible not to interfere in the cellular processes of integration and disintegration of a variety of proteins. The inhibition of the active protease in AIDS per se makes sense. However, the pharmacological administration of high doses of distinct aromatic substances is a non-physiological measure, connected with serious side effects which excludes their pharmacological use. Indeed, up to date, the pharmacological HIV protease inhibitors prove to be connected with side effects, which demand the absolute necessity of their replacements by phytotherapeutic mixtures. Apart from side effects such as kidney stones, damage of the liver, an increase of diabetic impairment of metabolism, CMV retinitis, and hemolytic anemias, these protease inhibitors, after a short-term administration, also demonstrate a loss of effect on the inflammatory process, which was misinterpreted as a result of an acquired resistance by HIV as well as an incompatibility with many drugs, especially with the ones of the group of cytochrome P450 inhibitors and inductors. 62. Nutritional possibilities in the prevention and treatment of AIDS. Looking at the formula of structure of the synthetic protease inhibitors, it becomes obvious that these are artificially produced aromatic compounds. As we have suggested lately, polyphenols as well as tannins and flavonoids are phytoprotective substances against harmful external influences. As aromatic substances, they cannot be synthesized by the animal organism. The nutritional supply of a variety of phytopolyphenols to the animal organism has the function of operating as a redox buffer and rebalancing oxidative stress conditions with their catabolic alteration of metabolism to the anabolic catabolic state of equilibrium. 63. Flavonoids and tannins are effective with respect to 1. Inhibition of lipid peroxidation, 2. Scavenging of oxygen radicals, 3. Binding and inactivation of pro-oxidative transition metals such as Fe and Cu, 4. Binding of proteins including attenuation of their enzymatic activity, protease inhibitors. Upon these reductive activities, flavonoids and tannins are oxidized themselves. A well-known example is the reduction of vitamin E by vitamin C or coenzyme Q. These mechanisms are at the beginning of a cascade of recycling. This example demonstrates that the multitude of almost 5,000 different flavonoids and tannins is used to overcome the oxidative state of the X-antioxidative molecules at the end of the cascade of recycling, by transferring it to a variety of native molecules. The stress-induced state of catabolic metabolism in AIDS is in the center of the pathogenesis. The correction of the connected whole body inflammations, caused by oxygen radicals and protease activation, is a compelling preventive and therapeutic action which urgently demands the use of phytotherapeutic polyphenol compounds. Possibilities and limits of treatment of hepatitis in anti-HIV positive individuals. Asymptomlessness and the stress-induced activation of liver inflammation in healthy individuals are characteristic of parenterally transmitted inoculation hepatitis, hepatitis B and C. The classic example for this occurrence is post-transfusional hepatitis caused by blood and blood products of clinically healthy blood donors. At the occasion of a study, made in the early 50s at the blood transfusion service of the Swiss Red Cross on recipients of lyophilized pools of mixed plasma of 50 to 70 healthy blood donors,
it was observed that this caused serious, sometime even lethal hepatitis in many ill recipients, 64 to 66. It is emphasized that in a contaminated organism with parenterally transmitted hepatitis inductors, now called hepatitis B and C, the aim of treatment has to be reduced to just reach a normal state of health. The administration of viricide, cytotoxic drugs is not able to eliminate these inductors from the organism. Proceeding from this knowledge, in Poland, for 2 decades, Bracowsko et al. Have been collecting data with the Tibetan prescription of phytotherapeutic formula, Padma 28, 67. They showed that this phenol-rich plant compound is able not only to reduce the serum level of hepatitis B antigens in hepatitis B patients but also to augment the serum level of hepatitis B antibodies. At the same time, an amelioration was observed in these patients regarding their clinical condition and the biochemical and histological results from their hepatitis. Based on these pioneer results, today, in patients suffering from chronically active hepatitis, the substitution with phytopolyphenolic mixtures has priority over other treatments. How does the nucleoside analogues treatment of AIDS patients influence their course of disease? After having examined 8 reports on HIV-positive long-term non-progressors, who stayed clinically symptomless for over 10 years, we realized that, without exception, they had not been treated with nucleoside analogues, 68 to 75. We consider this as a confirmation of our above-mentioned caution as to the prophylactic and therapeutic administration of these cell toxics, originally developed for treating cancer, in the autoimmune course of disease in AIDS. Nutritional supply of polyphenolic mixtures as basic treatment of anti-HIV positive individuals and AIDS patients. As initially showed, a positive anti-HIV test is an indication of an augmented formation of autoantibodies against cytoskeletal proteins, i.e. actin. This condition is pathognomonic for chronically active hepatitis. AIDS, a serious immunodeficiency syndrome, is the expression of a persistent hypercatabolic state of metabolism along with a stress-induced whole-body inflammation. A successful treatment of such conditions consists of the nutritional supply of a sufficient quantity of antioxidative and antiproteolytic phytophenolic mixtures, consisting of flavonoids and tannins. As neither the animal nor the human body are able to synthesize aromatic compounds, they are fully dependent on a sufficient supply of anabolic effective phytopolyphenolic mixtures, in order to adjust catabolic states of metabolism. These mixtures are present in drugs made of teas and spices. Padma 28 proved to be the most effective one. Additionally, it is recommended to balance other possible states of deficiency of vital nutritional components such as polyanions and essential fatty acids. Completing this review, we came across the publication by Etienne et al. Which remarkably emphasizes the insignificance of heterosexual intercourse in transmitting HIV. In this study, extended to 10 years, the authors say, male-to-female transmission was approximately 8 times more efficient than female-to-male transmission, and male-to-female per contact infectivity was estimated to be 0.0009. Obviously, AIDS is not a viral venereal disease, but an inflammatory autoimmune process. 76. End of article. Articles by Anthony Brink. Debating AZT. The Pope of AIDS. By Anthony Brink. When a doctor does go wrong, he is the first of criminals. He has nerve and he has knowledge. Sir Arthur Conan Doyle, 1859–1930, English author. Sherlock Holmes, In the Adventures of Sherlock Holmes. The Speckled Band, 1892, of Dr. Grimsby Roylott. For any number of obvious reasons, it would probably be disquieting to most folk at large to discover that Mother Teresa, to employ a fanciful illustration, had kept a Swiss bank account. One would imagine that the honesty of men working at the frontiers of science in that hazy twilight terrain between the known and the unknown, the certain and the speculative, would count for quite a bit as well. Particularly where their pontifications, advices, and theories have the potential both to reap magnificent honors and riches, and very directly affect us dumb fucks out in the laity, who sit at the feet of these guys and crave as much of their wisdom as we can get. And especially in a time of a perceived medical emergency,
or during the rise of an hysterical epidemic, fueled by a medieval fear of tainted blood and poisoned semen, and now evil mother's milk, in which we look up with frightened eyes to these secular sages for deliverance from tiny invisible enemies which we are told beset us. Mostly when enjoying our favorite evening recreation. Science at its outer limits is populated by no end of ambitious cowboys of modest acumen hungry for fame, glory, and the Ferraris in which some of their lucky chums in biotech cruise out of their labs parking lots in the direction of their Cessna hangars. They live so to speak in remote Wild West towns with lamentably few marshals to keep an eye on things. Many are the leftovers too mediocre to cut it in university environments who wind up in homes for scientific dullards like the politically powerful health bureaucracies of the National Institutes of Health and the Centers for Disease Control in the USA. As we've all seen, when these oracles mumble, press trumpets blare and the entire world eagerly gobbles up every word, without demur. Notwithstanding how many fake health crises they have delivered stillborn into the popular consciousness, like the idle herpes scare in the 70s, the great swine flu fiasco in the same decade, the phantom syphilis epidemic in the first half of the 20th century, and that shining emblem of medical idiocy, the pellagra plague in the U.S. South over the same period, treated inter alia with arsenic, electrocution, and ruthless quarantine, which turned out to be plain malnutrition among the politically awkward droves of poor white crackers in deep South industrial towns. We need contagious epidemics to fight. Even imagined ones. They're tremendously psychologically useful. Germ theory so dominates contemporary medicine that it seeks germs everywhere, the more virulent the better, and especially if they can be linked to our culture's great taboos, sex and death. Anything to avoid facing up to unappealing political realities like widespread chronic undernourishment among a shameful number of our countrymen as the time-honored and common-sense cause of broken health. Or, at the other pole, for those of us felicitously occupying the higher orders, factors inextricably tied to the excesses of our culture of affluence. Of course, the loftier the degree of scientific specialization, the sharper the point of the pyramid, the smaller and remoter the frontier town, and the fewer the guys with badges. As in a funny little corner of theoretical, some say virtual, virology called retrovirology, served at the commencement of the AIDS era by only a handful of labs run by the same guys who'd lost the war on cancer declared by Nixon in 1971, by putting all their money, and 40 billion of their countries, on the perfectly ridiculous theory that cancer was an infectious condition caused by viruses. Folk inclined to the view that a reasonable degree of personal integrity is essential to serve as a brake on the perennial temptation tickling largely unpoliced scientists at the frontiers of their specializations to make extravagant claims with fabulous commercial potential beyond those which their data really support might be put out to learn that the Pope of AIDS is a complete scumbag. We speak of Robert Gallo, who told the worried world at a press conference convened by the U.S. Health Department on April 23, 1984, before the publication of any paper for his fellows to assess, that he'd discovered the cause, a virus he said, of the poor health that a narrow subset of gay men with ruinous lifestyles were experiencing, later christened, in a flourish of conceptual surplusage, the acquired immune deficiency syndrome. Having sneaked through a patent application on the blood test he'd devised for his claimed viral culprit ahead of the previously lodged French one, thus guaranteeing him a fortune in royalties, Gallo went on to publish 4 papers in Science 2 weeks later. Then the trouble started, an exuberant international disputation over who stole the fake diamonds. For Gallo this was the Paula trouble that led to Monica. Luc Montagnier of the Pasteur Institute in France complained that the samples containing what he believed to be his newly spotted virus and which he'd trustingly sent Gallo had been flagrantly ripped off. He sued across the sea. Gallo brazenly countercharged his accuser. It was embarrassing for the U.S. administration to have its premier AIDS scientist accused of theft and fraud, but with the help of a gang of lawyers hired to fudge the facts and conceal boxes of crucial discoverable documents, Gallo got off, by dint of a neat political compromise agreeing a history, co-signed by no less than the presidents of the respective republics, Reagan and Chirac, in terms of which these two giants of modern biology were henceforth to be deemed co-discoverers of the AIDS virus. The sham began unraveling almost immediately. A troublemaking investigative journalist on the Chicago Tribune, John Crutzen, began sticking his nose in. He went to print with a comprehensively researched expose spilling the beans on Gallo's theft of Luc Montagnier's samples, even his photographs of them. Hardly able to do otherwise, Gallo's bosses in the National Institutes of Health instigated an inquiry with Yale biochemist Frederick Richards as overseer. Reviewing the 4 seminal research papers upon which the entire HIV/AIDS causation paradigm is founded, if feebly, the inquiry found fraud, a discrepancy between what had been reported and what had been done. The NIH watered it down, finding Gallo guilty merely of creating and fostering conditions that gave rise to falsified/fabricated data and falsified reports. This loyal whitewash was promptly criticized by Richards and by Senator John Dingell, who had got wind of the misfeasance in Gallo's laboratory, and had begun his own investigation under the aegis of his Subcommittee on Oversights and Investigations of the House Energy and Commerce Committee. The Department of Health's Office of Research Integrity reviewed the NIH report and disagreed with the cop-out. It had no trouble finding Gallo guilty of scientific misconduct, the gravest possible verdict, and a capital offense in career terms. So did the Dingell Committee in its draft report. Facing criminal prosecution for the perjury adorning his patent application, Gallo was forced to leave the National Institutes of Health in disgrace. On the scandal festered, until 1993, when happily for Gallo, it all went away. The government dropped the patent charges, and those of fraudulently making a misstatement in a scientific journal and failing to credit the work of other researchers in claiming it as his own. Why? Because a review board, comprising lawyers naturally, not scientists, had raised the bar in asserting a brand new revised definition of scientific misconduct, which Gallo's prosecutors in the Office of Research Integrity doubted they could clear. Unlike Sol Kersner, who kept his head down when the bribery case against him was dropped, Gallo boasted complete vindication. Before becoming famous for HIV, Gallo's laboratory had been found by an investigative panel of university scientists appointed in 1974 to be one of the worst offenders in the scandalous abuse of federal funds dished out during Nixon's war on cancer. 2 CO researchers later went down for embezzlement and taking secret gratuities. From this scientific cesspool was spewed the Constitution for the Terror, the founding papers of the most powerful, all-pervasive, and terrifying medical model of our time, the HIV/AIDS causation hypothesis. No wonder the Nobel Committee has set its face against the whole stinking shambles. Yet its integrity as a premise is assumed in the almost 100,000 papers in the subject that have been published since. Those critics making a living in the scientific establishment who point a finger at the emperor's pink arse do so at immense professional and personal risk, and for some, at terrible cost. But there's another story. Curiously, the Office of Research Integrity found that the fraud-tainting Gallo's claim to fame papers did not affect the validity of the paper's main conclusions, even though some of the key research work was described as of dubious scientific merit, and really crazy. Suffice it to say that others who have meticulously scrutinized Gallo's original HIV research claims, allowing for the purpose of reviewing them that the dubious research data is sound, have found them to be, well, shall we say troubling. The adventurous leap between the paper's contents and their headings, for starters. But that's another yarn still. Gallo's disgraceful behavior in relation to his AIDS research was no first. Had he not ascended to such power and influence within the federal health bureaucracy, it is likely that his claim to have found a single infectious cause for the disparate diseases grouped together as AIDS in the early 1980s would have been laughed out of court. After all, this was the bright spark who, with almost as much fanfare as that at his flashbulb-popping HIV press announcement, had loudly touted his discovery of what he claimed to be the first identified human retrovirus, HL23V, in the mid-70s. After another look, this exciting find turned out to be nothing of the kind, just an accidental laboratory artifact. His laboratory hadn't done the most basic controls. The virus had never existed. To his great embarrassment, Gallo had to retract his fancy claims, and HL23V then completely disappeared from the scientific lexicon. As the last misfired shots were going off in the failed cancer war, staged largely around the retroviral cancer hypothesis, and it had become irresistibly plain to everyone that cancer had nothing to do with germs, and the whole thing had been a monumental waste of money. Gallo and his mates, known in-house as the Bob Club, sought new funding opportunities for their imminently redundant laboratories. Ever eager to position himself where the action was, he began punting another retrovirus which he claimed to have discovered, HTLV1, as the possible cause of the odd diseases like Kaposi's sarcoma and pneumocystitis carinii pneumonia suddenly appearing to ail urban fast-track lifestyle gay men in San Francisco and New York. The virus had in fact been identified by biologists in Gallo's lab, principally Poise and Russetti, not Gallo, but true to form he appropriated the discovery and took the accolades. Wanting the virus to be all things, the theory that HTLV1 could be responsible for AIDS was ludicrous. He had previously claimed this virus, again on absurd grounds, to be the cause of a rare form of leukemia, which amounts to disorderly immune cell replication, not premature cell death. One of the most exciting stories of 20th century biology, he gushed. Nobel laureate Kary Mullis thinks it a joke. The virus had first been posited to be a cell division stimulant, not a killer. Obviously, Gallo's new converse role for HTLV1 went up like a lead balloon, but it didn't matter, because it wasn't long afterwards that Montagnier sent Gallo his samples, and we know the rest. In cravenly seeking the imprimatur of big American science, by seeking the endorsement for his work of an abject rogue, Montagnier naively left his keys in the ignition, and the next thing it was gone. Gallo resprayed Montagnier's lab as HIV. It was later renamed HIV, the human immunodeficiency virus, on the basis of Gallo's claims, without proof to warrant its fearsome title. Unless one thinks that correlations disclose proofs of causation. As if sparrows sometimes seen on telephone cables cause crossed lines. Whether HIV, or rather the minute biological traces said to evidence its presence, actually lives up to its frightening billing, is something Gallo can't seem to make up his mind about. This ought to come as some comfort to those who live and wait for the clatter of the hangman's key. Once insisting that HIV kills like a truck, and would kill Clark Kent, he now concedes, we don't know that, 100% of people infected with HIV will die with AIDS. We don't know that. We shouldn't be predicting that, and it could even precipitate suicide. They shouldn't have put that on the front page of the Washington Post, even if it were true. But the fact is that we just don't know. In 1995, the Pasteur Institute's Simon Wayne Hobson confessed, an intrinsic cytopathic effect of the virus is no longer feasible. The biggest medical research effort in history has found HIV to be biologically inactive. Gallo has tried weaseling out of the difficulty created by this humbling observation by suggesting that cofactors might be involved in AIDS, since HIV can't do any mischief on its own. Time magazine's 1996 Man of the Year, David Ho's opposite assertions in 1996 have imploded on his childish mathematical errors. Gallo had lots to say about a virus called HHV8 for a while, implicated as a cofactor in the development of that signal AIDS condition Kaposi's sarcoma, but like all other exciting breakthroughs in AIDS research, it too has proved to be just another flash in the pan. Worse still, it is now generally accepted, and since 1994 by Gallo too, that those horrible skin blotches have nothing to do with HIV at all. At last count, Gallo was on SABC TV a couple of years ago, singing his own praises for his alleged breakthrough anti-HIV protein half, distilled from the urine of women with child. About which we have heard nothing since. Naturally, since it was just another rodeo stunt. Gallo's new laboratory in Baltimore had produced nothing to show for the millions he had duped state and municipal authorities into giving him, and was about to have its plug pulled by the Maryland legislature accordingly. A neatly timed very important discovery defeated the danger. Since the case for Gallo's HIV/AIDS hypothesis is invariably pressed with calls to the authority of its famous protagonist, in the absence of scientific proof in the sense that most curious folk understand, it's as well that we know what kind of bloke we're relying on. With such scintillating credentials as Gallo's, no wonder that astute German virologist Stefan Lanka, referring to HIV/AIDS, Luc Montagnier, and Gallo, talks of a medical theory concocted by a French mediocrity who right from the start doubted the validity of a virus-only theory of AIDS causation, and only last week unleashed a new wave of doubt, and an American scientific gangster who had committed so many crass, self-aggrandizing blunders in the previous decade, that he could not really be relied upon to tell the time correctly. The Einstein of modern biology, Kary Mullis, doesn't mince words either. He considers Gallo and his acolytes so stupid they're to be pitied. End of article. Debating AZT. How could they all be wrong? Doctors and AIDS. By Anthony Brink. I suppose one has a greater sense of intellectual degradation after an interview with a doctor than from any other human experience. Alice James. A response sometimes heard to the expression of doubt about the integrity of the HIV/AIDS paradigm as a medical model for understanding disease incidence is, how could all the doctors in the world be wrong? There are many possible answers to this question. One might point out that unanimity has never guaranteed the soundness of medical constructs, and examples of this abound. The history of medicine, both ancient and modern, is a wrecking yard full of broken and abandoned ideas. In this century alone, innumerable medical theses have collapsed to which nearly all doctors once subscribed, such as bacterial theories of scurvy, beriberi, and pellagra, and more recently, the immunosurveillance and retroviral theories of cancer etiology, for which billions of dollars funded thousands of convincing research papers during the War on Cancer declared by Nixon in 1971. Then there was swine flu. 1976 saw President Gerald Ford on television, at the behest of the American medical establishment, solemnly urging all Americans to get vaccinated against an imminent deadly influenza epidemic. About 50 million Americans were panicked into being immunized with useless or harmful vaccines rushed onto the market. Adverse reactions resulted in damages claims of $2.7 billion. Not a single case of swine flu appeared subsequent to the death of a sick recruit undergoing basic training in a boot camp in New Jersey, hardly an unusual event, that had ignited all the hysteria. Before HIV/AIDS, and alongside the mad cow craze in Britain and the avian flu folly in Hong Kong, the Great Swine Flu fiasco was perhaps the most telling instance in recent times of how medicine can lose its head. Another answer to the question goes to the fact that most doctors have scarcely more than a layman's grasp of the concepts that populate biology at its molecular horizon. For instance, most would gape dully if asked to define the peculiar characteristics of a retrovirus, like HIV, we're told, as distinct from other viruses, or distinguish endogenous and exogenous retroviruses, or articulate the rival contentions advanced by molecular biologists about whether the whole of retrovirology might be a mistake, a wrong turn at a scientific road fork, a bad inference drawn from the evidence of certain metabolic biochemical phenomena which look odd when seen against old-fashioned rules of molecular genetics, and the possibility that retroviruses might not exist as infectious agents at all, that it is rather the classical dogma that needs an overhaul. Taxed about the HIV theory of AIDS, most doctors can do little more than quote the claims of their authorities, like priests citing papal bulls and encyclicals, making obeisance to their cardinals. A third answer would make the impudent point that it is fallacious to imagine that doctors generally have a superior capacity for reasoning than their patients. The notes given medical students speak to the scant education that doctors receive in this art. To read them is to see how flimsy medical and biological theories are dished up as fact for rote learning, making the kind of call-and-answer instruction one sees in farm schools in this country look like an adventure in lateral thinking training. Doctors do so well at school because they're the kind of guys who are the most easily schooled in myths and legends to outdo the Hare Krishna people. Especially virologists, who occupy the haughtiest medical echelons, but who seem to have the dimmest bulbs in the upper story, as revealed by what they swallow without a hiccup, and regurgitate to their students. Like the timeless French fancy, le rage, that a bite from a dog acting wild and crazy can make you go mad too, and die. But not the dog, man is the end host, they say. You can go the same way from eating steak. Although nobody can plausibly say why. Or some cancers are caused by viruses and are infectious. Or the most hilarious notion of them all, having sex can be deadly. Mother's milk too. But not spit. All of a sudden. After millions of years. Thanks to a mutated virus from monkeys. Or maybe the moon. And all of this without any evidence. Not a shred. And there's a funny part to it. You might be feeling fine. But you're sure to go in 6 months' time from any one of a couple of dozen diseases or malignancies. No, make that 2 years, well actually 5, shall we say 8 or 10 or 12, maybe 15, okay perhaps your life is just shortened a bit. Definitely? Yes, most certainly, no, not necessarily. Look, we don't know. How, why? We don't have the faintest idea. Theories zigzag like a drunk at the wheel. We are still confused, but at least now we are confused at a higher level of understanding. Harvard Medical School Professor of Immunology Paul Johnson, excuse me. Is this the circus? Nor do doctors necessarily proceed from a more rational mindset than Joe Public does. The opposite may be the case. That HIV/AIDS as a medical construct could have taken root so richly among doctors, despite its absurd fundamental tenets, which fly in the face of everything known to virology, illustrates the point. As Harvey Bialy, scholar in residence at the Biotechnology Institute at the University of Mexico and editor-at-large of the prestigious science journal Nature Biotechnology puts it, the HIV theory of AIDS turns immunology upside down and inside out. To begin with, never before was the presence of antibodies taken to be prognostic of future disease. They used to be thought of as good things evidence, where the patient appears healthy, of a successful immune response to a pathogen defeated. Former molecular biology professor at Johns Hopkins and Harvard Universities, Charles Thomas predicts that after the balloon pops, historians will be studying the flight of common sense in the lunacy of the AIDS age, for 100 years, how America gave AIDS to the world. But since HIV/AIDS as a diagnostic construct is still hegemonically regnant in our time, the point about the way doctors as a group tend to think needs illustrating with a different example. What better than the turn medicine took during the Third Reich? The Nazis' virulently irrational and barbarous doctrines of racial hygiene found huge appeal for German and Austrian doctors in that era. No other profession was as well represented on Nazi Party membership lists. From an ostensibly sober, rational profession functioning as an elite caste in a culture that seemed itself to be the fruit of the Enlightenment, just under half of them were card-carrying Nazis. Of course not all engaged in the sadistic butchery of Untermenschen for which the Nuremberg doctors' trials were conducted, but it would be a mistake to imagine that such criminals were aberrant quacks from the fringes, flourishing like vermin on the opportunities created within the Nazi eugenics paradigm. In fact, many medical practitioners and academics tried or named in testimony at the trials had enjoyed international eminence in their professional fields. Dr. Edwin Katzenellenbogen, for instance, who got life imprisonment, had served on the faculty of the Harvard Medical School. Scholars of religious thinking have long known that the more horrible and improbable the founding superstitions of a new faith, the greater its capacity to mobilize the popular imagination and the stronger the force of its revolutionary engine. In medicine, religion's first cousin, the same sometimes applies. Like an infant upstart religion with imperial designs, the HIV/AIDS paradigm calls for a vigorous rebellion against long-established models of understanding. Woe betide any conservative scientists reluctant to become conversos to the rude new creed, who point out that the new theory is absurd on its face, that the link between AIDS and sex is no stronger than its link with sleeping. They become marginalist like Jews defying the demands of medieval Christendom, not racked and burned, but ostracized, scientifically defrocked, blacklisted and delegitimated, stripped of research funding, banned from lecturing podia, kicked out of their laboratories. Rendered unemployable in academia or industry, menaced with confinement in psychiatric wards, isolated from graduate students in whom they might instill similar heretic doubts, and barred from publishing in the journals that once craved their papers. But naturally, radical political dissident Noam Chomsky, professor of linguistics at Massachusetts Institute of Technology in the U.S., has pointed out that if you serve power, power rewards you with respectability. If you work to undermine power, you are reviled, imprisoned, driven into the desert. The AIDS phenomenon at root is a vast pumping aggregation of interests with enormous political and economic power. Doctors and scientists who challenge its sacred tenets risk attracting the wrath of the revolution's Red Guards. They won't be thrown from windows, but their careers will be over. For their reactionary intransigence, these critics will be marked always with pejorative epithets, as persistent as tattoos, like discredited, loony, maverick, dangerous and irresponsible and pernicious. Just to make sure we correctly tell the wits from the dunces. And to discourage us from asking, well, what are these guys actually saying? A fourth explanation lies in the fact that for all their social status and prestige, in truth doctors generally function close to the bottom of the food chain in the medical-industrial complex, and serve as little more than a thoughtless delivery system for the pharmaceutical corporations whose wares they peddle makes the medical drug industry one of the most profitable legal enterprises on the planet. Just how little room doctors are allowed for independent judgment founded on their own observations is revealed in the fact that in some places a doctor who declines to follow an approved treatment regimen such as chemotherapy for cancerous tumors, in view of his empirical assessment of its utter uselessness and lethal toxicity, risks sanctions from his controlling guild. Imagine the trouble a doctor would be in were he brazenly to announce his conclusion that having investigated the business, reactive HIV antibody test results are virtually meaningless, pointers to no more than heightened nonspecific immunologic activity. And were he to refuse to diagnose negative or positive, selecting for life or death, like a Nazi doctor calling links or Rex, or marking plus on the medical files of slow or crippled German children, to mark them for murder during the euthanasia program. In sum, one doesn't have to cast about too far for answers to the question, how could all the doctors in the world be wrong about AIDS? Medicine's penchant for screwing up magnificently, its characteristic intellectual sluggishness, and the appeal of magical thinking for its practitioners is plain to anyone who turns back a few pages. End of article. AIDS in Africa. A Case of Racism vs. Science. By Rosalind Harrison-Chirimuuta and Richard Chirimuuta. AIDS in Africa and the Caribbean 1997. Introduction. Western scientists have promoted the hypothesis that the AIDS epidemic began in Africa, arguing that either AIDS had existed for many years in an African lost tribe or that a retrovirus crossed the species barrier from monkey to man. The scientific evidence in support of this hypothesis has included AIDS-like cases from Africa that predated the epidemic in the West, seroepidemiological evidence for early African infection, and the isolation of retroviruses from African monkeys considered similar to the human immunodeficiency virus. Yet when the scientific literature supporting an African origin is examined, it is found to be contradictory, insubstantial, or unsound, whilst the possibility that AIDS was introduced to Africa from the West has not been seriously investigated. The belief that the AIDS epidemic originated in Africa has also distorted Western perceptions of the scale and mode of spread of the epidemic in Africa, and it would seem that much of the research into AIDS in Africa has been influenced by racism and not science. The acquired immune deficiency syndrome, AIDS, was first recognized as a clinical entity in 1981 in the United States, and although the majority of cases even today have been reported from the United States, the Western scientific community has convinced the world that it is primarily an African disease and an African problem. To explain how a disease originating in one continent was yet disseminated to the rest of the world from another, the scientists have argued that there was a remote Central African lost tribe in whom the virus had been present for centuries, or alternatively who acquired the infection from monkeys 30 or so years ago, Haitians, but no one else, working in Central Africa then became infected, presumably heterosexually, and, on returning home, spread the disease to homosexual American tourists. 5,6 pages 17-20. By this circuitous route the virus reached the United States and from there spread to the rest of the world. Because we suspected a racist motivation for the science that was arguing for AIDS from Africa, we decided to review the scientific literature, eventually publishing our work in a book, 7. When questioning the African hypothesis, we anticipated a difficult task, as the research was conducted by reputable scientists and was subjected to peer review prior to publication. As our study progressed, it became increasingly clear to us that the racist preconceptions of the researchers led them to conclusions that had no scientific foundation. The ideology of racism. It is perhaps unwise to assume a consensus view of racism where none may exist, and for our purposes we would consider racism to be the ideology promoted initially by the Caribbean sugar planters and slave merchants to justify, sustain, and defend their activities so important to the enrichment of Europe during the 17th and 18th centuries. The ideology was adapted and developed during the period of European colonization during the 19th century and in the 20th century, reaching its apogee in the death camps of Nazi Germany. Unlike the variety of superstitious beliefs Europeans held of other peoples in previous centuries, racism was relatively systematic and internally consistent, and with time acquired a pseudo-scientific veneer that glossed over its irrationalities and enabled it to claim intellectual respectability. 8. Although the edifice of racism has begun to crack in the latter part of this century, racism remains integral to the European worldview. Many leading doctors and scientists of their day made their contributions to the pseudoscience of racism. 8,9. When humans were placed at the top of the evolutionary tree, Africans were allocated a separate species between other humans and apes, and there were numerous suggestions that Africans had sexual intercourse with apes, or were the result of such unions. As Africans were deemed more akin to animals than humans, they were by definition incapable of civilized behavior. They were believed to be sexually unrestrained and to have larger sexual organs than other races, and were therefore more prone to sexually transmitted diseases. They were deceitful, treacherous, lazy, faithless, cruel, and bad-tempered. African skulls were studied and were considered to be smaller than those of Europeans, establishing beyond doubt that Africans had the lesser intelligence. 8. In one form or another, explicitly or implicitly, many of these notions have appeared in the scientific literature about AIDS and Africa. Racism and AIDS from Africa. The first black people diagnosed as suffering from AIDS in any number were Haitians living in the United States. 10,11. The possibility that they may have caught the infection from Americans in the United States or in Haiti was not given serious consideration, and Haiti was immediately accused of being the source of the epidemic. 11. Soon Haitians with AIDS were being reported from all over the Western world. 12, 13, 14, 15, and the Centers for Disease Control, CDC, in Atlanta, Georgia, included Haitians as a group at risk for AIDS along with homosexuals, intravenous drug users and hemophiliacs. It was only in 1985 that CDC, faced with overwhelming evidence that Haitians per se were no more at risk for AIDS than anyone else, 16, removed them from the high-risk classification, but not before Haitians en masse were dismissed from their jobs, evicted from their homes, and even housed in separate prisons. Abandoning Haiti, the researchers then turned their attention to Africa. One of the reasons given by scientists for this turn to Africa was the high incidence of Kaposi's sarcoma, KS, in Africa, although it was clear from the beginning that the benign clinical course of African KS was very different from the aggressive, disseminated form of KS in AIDS patients. 17. A number of AIDS-like cases were reported retrospectively, the most cited being a Danish surgeon who worked in Zaire and died in 1977. 18. This patient was given prominence in the best-selling book by Randy Schiltz and the band played on, where, under Dramatis Personae, she is listed as Danish surgeon in Zaire, first Westerner to have died of AIDS, and is described in the following manner. The battle between humans and disease was nowhere more bitterly fought than here in the fetid equatorial climate, where heat and humidity fuel the generation of new life forms. Here, on the frontiers of the world's harshest realities, Greth Rask tended the sick. 19. Jonathan Mann, former director of the AIDS program for the World Health Organization, WHO, and medical textbooks are now citing the case as evidence that AIDS originated in Central Africa. 20,21, it was claimed that she acquired the infection from her patients, at least one of whom had KS, but there was no firm evidence that she died of AIDS, and other diagnostic possibilities were not considered. In 1988, 5 years after the case was published, we learned that her serum had been tested and found human immunodeficiency virus, HIV, enzyme-linked immunosorbent assay, ELISA, negative, 22, but the author of the original paper has not published this information in the scientific literature. Although such AIDS-like cases are presented as evidence that the human immunodeficiency virus existed in Africa prior to the American epidemic, only African cases are considered and the many instances of AIDS-like cases documented in Europe and America are conveniently ignored. 23. Indeed, on the opposite page to the report of the Danish surgeon in the same issue of The Lancet was an account of an AIDS-like illness in a young German homosexual. 24. But whilst non-AIDS in a Danish surgeon heads the citation index proving an African origin, the German case has been completely ignored. The next source of support for the African hypothesis came from the seroepidemiological studies undertaken in Africa or on African serum stored in the West. This research, more than any other, has been at the foundation of all the fantastic stories of millions dying in Africa. Using an enzyme-linked immunosorbent assay, seropositive figures of 25% of patients attending a clinic in rural Zaire in 1984, 25, 50% of the Turkanas in Kenya from 1980–1984, 26, and 66% of children in Uganda in 1972, 27, were reported. As AIDS was rare or unknown in the areas where the serum was collected, one would expect the authors to have had serious doubts about the reliability of the tests, but sadly scientific skepticism has never been a feature of AIDS research in Africa. One of the most cited studies was undertaken on serum collected in Zaire in 1959, 28, using a number of tests in addition to ELISA. Only one of 1,213 plasmas was positive, but the identity of the donor, described as rural Bantu, was no longer known. As with the sporadic AIDS-like cases, only seroepidemiology in Africa is considered relevant to the question of the origin of HIV. A study using the same tests was undertaken on stored serum taken from Aboriginal Amazonian Indians in Venezuela in 1968–69, and 9 of 224 samples were positive on all the tests. 29. The results were challenged by other researchers as probable false positivity. 30. But the single positive sample from Zaire continues to be cited as evidence that the World AIDS epidemic began in Zaire 35 years ago. In an interview shown on British television, Professor Hans Mann, head of Virology and Immunology section and Professor of Medicine at the West German Center of Primate Research at Göttingen, discussed the problems of seroepidemiology. We had conducted quite extensive experiments in respect to the epidemiology of the first human retrovirus, HTLV human T lymphotropic virus I. For this reason we had several thousand serum samples frozen and saved in our refrigerated stock. When the news came that there was another, and new human retrovirus discovered, the AIDS virus, we could immediately search among our stock and probe for an earlier presence of this virus in Africa. These tests quickly and clearly gave results, namely, that the first positive probes which we could find among our more than 7,000 serum samples are dated only after the beginning of the 80s, from the years 1982 to 83, and that among samples from before that date, and we had quite a lot of that earlier time in our stock, not a single one proved positive. We have concluded from all this that most other researchers had probably fallen victim to the technical difficulties connected with the conservation and analysis of older serum samples. And the American authors who originally had produced those high percentage data had to correct them, but certainly, once some wrong information like that has been put into circulation, it continues to go on. This has led to quite a lot of friction between some African states and the United States. 31. Later in the same interview, when asked why AIDS is not considered to have originated in the United States, Professor Hans Mann made the following comment. Testing of the kind being done in Africa and to that volume has never been done by anyone in America. Nobody has looked at the stocked blood serum in the USA, and there certainly is much more there than in Africa. Nor has anyone asked what happened to the general population. Only one single group, the homosexual community in San Francisco,
has been analyzed and the results showed a high percentage of HIV positivity already by the mid-1970s. But no other samples have been tested to the extent done in Africa. I think this should be clearly said. 31. Why, then, if this research is valid, and there have not been any serious criticisms, have other AIDS researchers persisted in arguing that the African AIDS epidemic preceded the epidemic elsewhere in the world? And if the tests are unreliable, why are the predictions that millions of Africans will soon die from AIDS still presented without comment? How, indeed, is it possible that a virus could spread so much more rapidly by heterosexual contact in Africa than anywhere else in the world? It is here, as in so many other aspects of AIDS research, that racist beliefs about the sexual propensities and promiscuity of Africans conflict with scientific evidence, and in such a confrontation belief is almost invariably the victor. Researchers had originally proposed that AIDS was an old disease of Africa that had reached the West via recent intercontinental travel. 3. A rather curious notion given the enforced intercontinental travel of up to 100 million Africans in previous centuries. 32. As this hypothesis became increasingly untenable, attention was diverted to the possibility of a monkey origin of the virus. Such ideas cohabit easily with racist notions that Africans are evolutionarily closer to subhuman primates. Dr. Robert Gallo and his co-workers were among the pioneers of this line of research, both for HTLV-I and HTLV-3, later renamed HIV. 533-34, two of Gallo's colleagues, Kanki and Essex, reported the isolation of a virus similar to HTLV-3 in macaque monkeys who were suffering from an AIDS-like illness, and labeled it simian T lymphotropic virus type 3, STLV3, of macaques. 35. For those who were arguing an African origin of the AIDS virus, an Asian monkey like the macaque was not a suitable source, but less than 6 months later the same researchers reported finding the virus in wild-caught African green monkeys from Kenya and Ethiopia. 36. This research, like most other research on AIDS in Africa, was motivated only by a desire to prove an African origin of the disease, and was greeted with enthusiasm by the Western scientific community. Discussion quickly moved on to the question of how the virus crossed the species barrier, and 2 AIDS experts from St. Mary's Hospital in London even offered this explanation. Monkeys are often hunted for food in Africa. It may be that a hunting accident of some sort, or an accident in preparation for cooking, brought people in contact with infected blood. Once caught, monkeys are often kept in huts for some time before they are eaten. Dead monkeys are sometimes used as toys by African children. 37. Are we seriously to believe that African parents are so desperate for toys for their children that they give them putrefying carcasses of dead animals? More fantastic suggestions were published in The Lancet. Sir, the isolation from monkeys of retroviruses closely related to HIV strongly suggests a simian origin for this virus. Several unlikely hypotheses have been put forward. In his book on the sexual life of people of the Great Lakes area of Africa, Kashamura writes, Pour stimuler intense, on leur inocule dans les cuisses, la région du pubis et dos du sang prélève sur un singe, pour un homme, sur un ganon, pour une femme, to stimulate a man or a woman and induce them to intense sexual activity. Monkey blood for a man or she-monkey blood for a woman was directly inoculated in the pubic area and also the thighs and back. These magic practices would therefore constitute an efficient experimental transmission model and could be responsible for the emergence of AIDS in man. 38. This came in for particular derision at the Conference on AIDS in Africa held in Naples in October 1987. When queried regarding the plausibility of a premise put forth in a letter to The Lancet suggesting that a bizarre tribal ritual of injecting monkey blood into the pubic region of young African men and women to stimulate intense sexual activity could be responsible for the emergence of AIDS in man, researchers from Zaire, Congo, and Belgium were unanimous in declaring it to be preposterous. 39. It is hardly surprising that Western AIDS researchers have become persona non grata in many African countries. Most Africans, in fact, have little contact with monkeys. 40, and amongst those who regularly hunt monkeys, for example the pygmies of the equatorial rainforests, AIDS is notable for its absence. 41. On the other hand, in recent years there has been a marked increase in contact between man and monkeys, not in Africa but in the West. In the 1920s the transplantation of monkey testes to humans was widely practiced, and many thousands of men in Europe, America, and Australia received the benefit of this operation that promised to restore their youth and vigor. 42. Monkeys have been used widely for scientific research, and with the discovery that their kidneys provide excellent tissue culture material for virus isolation, propagation, and vaccine production, hundreds of thousands have been caught and transported from their native haunts. 43. If there is any truth in the hypothesis that HIV originated in monkeys, and African monkeys are not the only candidates, it would seem more appropriate to investigate modern medical research than speculate about the customs and behavior of Africans. Although the African green monkey hypothesis was widely accepted, it came under increasing scientific challenge. Attempts to repeat the Essex and Kankakee experiments on other wild African green monkeys were unsuccessful. 44. And the genetic sequences of the virus isolated from laboratory macaque monkeys, the virus Essex and Kankakee claimed to have isolated from wild-caught green monkeys and another supposedly human virus called HTLV4, were found to be identical. 45. Essex and Kankakee were then obliged to admit that their green monkey virus was a laboratory contaminant. 46. A retrovirus was eventually isolated from African green monkeys, but it bore little resemblance either to the macaque virus or the human AIDS viruses, and could not have originated from African green monkeys in recent times. 47,48. It is difficult to understand why this virus has been called simian immunodeficiency virus of African green monkeys, SIVAM, as it does not cause immune deficiency. In all this confusion of viruses, one question surely needs to be asked: what is the origin of the virus that caused AIDS-like illnesses in laboratory macaque monkeys? This virus does not occur in wild macaque monkeys, but does have some similarity with the human AIDS viruses. Had these monkeys been subjected to experiments with retroviruses, and did the appearance of AIDS-like illnesses in the monkeys predate the human AIDS epidemic? It is instructive for anyone who still has illusions in the objectivity of science or the integrity of some AIDS researchers to read the October 1988 edition of Scientific American. The issue was devoted to AIDS, and the section titled "The Origins of the AIDS Virus" was written by Essex and Kankakee and was illustrated by a full-page color picture of an African green monkey. 8 months after admitting that the African green monkey virus was a laboratory contaminant, Essex and Kankakee have the audacity to state why SIV is endemic in these wild African monkeys but seems to do them no harm, and is also found in the captive Asian macaques, where it causes disease, was, and still is, an enigma. 49. Does this representation of discredited data signal the abandonment of any pretense of scientific integrity in order to promote conscious and deliberate propaganda? Other attempts to implicate Africa in the AIDS epidemic also came to grief. Dr. Anthony Pinching and his team from St. Mary's Hospital, London, claimed that a particular genetic variant, the GC1F allele, predisposed the person to infection with HIV, and that this variant was common in Central Africa. 50. The GC1F allele had, in fact, been found to be common in the Bai aka Pygmies of the Central African Republic and the Pol Fula of Senegal, ethnically distinct groups in whom AIDS was either rare or notable for its absence. 41. But it would seem to European minds all Africans are the same. This research was reported in the media as a major breakthrough in the search for a cure for AIDS. 41. But a year later, after a number of other laboratories failed to confirm the findings, Dr. Pinching admitted that their original data was erroneous. 51. At least Dr. Pinching, unlike Dr. Bigberg and so many other AIDS researchers, had the courtesy to admit his error publicly and apologize to his fellow scientists for the extra work he had caused, although his apologies were not extended to the many Africans whom he had offended. Although many AIDS researchers now appreciate that they have offended and angered many black people, they remain ignorant of their unconscious racism and continue to give offense. The September 1988 edition of Medicine International was devoted to the subject of AIDS, and as usual there was an article on AIDS in Africa, but no similar discussion about AIDS in any other continent. The authors commented on the problems created by earlier AIDS research in Africa. Initial claims that the disease had been present in Africa for long enough for widespread immunity to have developed in exposed populations were false. Epidemics of AIDS were as new in Africa as elsewhere. Considerable damage has been done to international research collaboration as a result of these claims. 52. But later in the same article, the scale of African AIDS epidemic has led to speculation that heterosexual transmission is more efficient in Africa than elsewhere. Social and cultural factors, such as the African tradition of male sexual freedom, may also play a part. The circulation of myths such as the only cure for AIDS being to have sex with a virgin is likely to have a greater effect on transmission in Africa than in developed countries. 52. What do the authors of this paper know about African traditions of male sexual freedom, and does no such tradition exist in the West? And on what evidence are we to believe that a significant number of African men are having sex with virgins to cure themselves of AIDS? But then if you already believe that Africans are more primitive and superstitious, no evidence is required. Other AIDS researchers have recognized that their past activities have caused problems. The British Medical Bulletin of January 1988, titled AIDS and HIV Infection, The Wider Perspective, was edited by three notable exponents of the African connection, Anthony Pinching, Robin Weiss and David Miller. They provide a classic example of muddled racist thinking. In the case of some early studies in Africa, techniques were used that had not been sufficiently well validated for African sera, given the prevalent hypergammaglobulinemia and a notorious tendency to stickiness and false positive reactions in antiglobulin assays. The observations derived from these studies have led to some confusion and have also tended to damage the credibility of foreign scientists working in Africa, especially among local leaders. 53. Who was confused by this bad science? Certainly not Africans, whether ordinary citizens or local leaders. The racist themes were all too familiar, the response was anger and not confusion, and the discrediting of the science came as no surprise, as it was never believed in the first place. The AIDS experts continue. Additional problems have been created when investigators have spent a short time collecting sera and basic data in a developing country, often with little guidance from local investigators, and then published the data without reference to the original context. This has tended to produce scientific data that has not been adequately placed in an anthropological perspective. 53. In other words, data collection was biased or inadequate, and this led to a misinterpretation of results. The racism responsible for this is charmingly described as an inadequate anthropological perspective. But worse is to come. Even worse, it has led to denial and resentment, jeopardizing essential and potentially fruitful collaboration between investigators in the developed and developing world in a study of an issue of mutual concern. This has been particularly damaging when the pursuit has apparently been the origin of AIDS and HIV, an essentially academic question, however interesting. Such investigations have often been taken to imply blame on the region that appears to be the source. Although they were certainly never intended to impugn any community in this way, it is not difficult to see how such perceptions arose. 53. Recognition that the faulty techniques described at the beginning of the paragraph provided the evidence for an African origin for HIV is beyond the wit of these clever scientists, who then accuse Africans of denial and resentment when they refuse to accept their findings. Let us gratefully accept their condolences for the injuries they have inflicted, and put aside our resentments, so that we can leave ourselves open to more of the same, to be found later in the bulletin. HIV infection appears to have spread over much of the world during the decade 1976 to 1986, mirroring on a large scale the spread of its most obvious predecessor, syphilis, in Europe in the 1490s. As with early syphilis, the international spread of AIDS has led to a process of attribution and denial about the origin of the disease. However, it seems most likely that HIV spread to the United States from Africa, perhaps via Haiti, in the mid-1970s and from the United States to many Western countries in the late 1070s and early 1980s. 54. Others are not so confident,
at least when they address Africans at AIDS and Africa conferences. Luc Montagnier, the first scientist to isolate the virus that causes AIDS, agrees that if an isolated population in Africa existed as a reservoir for the virus, researchers would have found it by now. The data suggesting that the virus comes from Africa are weak, Montagnier said. Maybe we should look to another part of the world. 55. Jonathan Mann, then the director of WHO's AIDS program, also felt obliged to distance himself from an African origin. The World Health Organization's position is that there is not yet enough information about the origin of the virus. There are absolutely no data to support any hypothesis. The more information that emerges, the less we know about where this virus came from, how long it has been in the world, and how it grew to become the problem that it is today, he said. The syndrome has too often unveiled thinly disguised prejudices about race, religion, sex, social class, and nationality, and the Africans properly resent that Africa has been singled out, Dr. Mann said. If San Francisco was accused of being the original source of HIV with no more proof than there is that Africa is the source, special interest groups would be up in arms, he said. Dr. Mann said that nothing will keep people from coming up with cheap hypotheses about the origin of AIDS. They die a natural death when no subsequent evidence develops to take them seriously. But journals should have a special page for them labeled fuzzy ideas, he said. The real danger is that future authors might use such discredited, but published, hypotheses as scientific references for future articles, he said. 39. This would seem a classic case of white man speaking with forked tongue, as there is a qualitative difference between racism and mere fuzzy ideas, and whilst the publication of fuzzy ideas may be no more than a reflection on the quality of the journal, racism should find no place in its pages. The director of the WHO's AIDS program and his associates were in a position to request that the medical and scientific journals adopt and implement anti-racist policies. Instead, they were content to show their bleeding hearts only at conferences attended by Africans. Although racism can be found in abundance in the medical literature about AIDS and Africa, two psychologists, J. Philip Rushton and Anthony F. Bogaert, have drawn together these ideas and have attempted to give them a pseudo-scientific coherence. According to the British newspaper The Independent on Sunday, Rushton has received funding from a racist American trust and was investigated by the Canadian police under the hate propaganda laws. Rushton and Bogaert's paper, titled Population Differences in Susceptibility to AIDS, an Evolutionary Analysis, was published in a leading British journal, Social Science and Medicine. The abstract is as follows. Previously we have reported population differences in sexual restraint such that, higher socioeconomic status lower socioeconomic status, and Mongoloids Caucasoids Negroids. This ordering was predicted from a gene-based evolutionary theory of R/R reproductive strategies in which a trade-off occurs between gamete production and social behaviors such as intelligence, law-abidingness, and parental care. Here we consider the implications of these analyses for sexual dysfunction, including susceptibility to AIDS. We conclude that relative to Caucasians, populations of Asian ancestry are inclined to a greater frequency of inhibitory disorders such as low sexual excitement and premature ejaculation and to a lower frequency of sexually transmitted diseases including AIDS, while populations of African ancestry are inclined to a greater frequency of uninhibited disorders such as rape and unintended pregnancy and to more sexually transmitted diseases including AIDS. 56. It is not possible to discuss this article in detail, but the only difference in substance from the pseudo-scientific racism of previous centuries is the different ranking order of the races. Mongoloids are now superior to Caucasoids, although Negroids, of course, remain at the bottom. Meaningless algebraic presentations such as R/K only give a modern veneer to very old ideas. We are told, for example, that the average cranial capacity of Mongoloids is 1,448 cubic centimeters v 1,334 cubic centimeters for Negroids, whilst genitalia and secondary sex characteristics of Mongoloids are, of course, small and that of Negroids large, and for such reasons AIDS is rampaging through Africa. It is difficult to believe that such an article would be published anywhere but in a right-wing fringe magazine, but after a decade of AIDS pseudoscience anything seems possible. The AIDS establishment has typically responded to the charge of racism with the counter-accusation that such criticism deny an African AIDS epidemic, giving African governments an excuse not to take measures to contain the epidemic. In fact, we do not deny the existence of an AIDS epidemic in Africa and elsewhere in the world, but believe the scale of the epidemic is open to question. Whilst doctors from the West claim there are tens of thousands of Africans dying from AIDS, and that millions are already infected with HIV, the experience of African doctors and ordinary people is very different. One Zimbabwean woman who in 1988 had not seen or heard of anyone with AIDS said that it was like being asked to believe in the Holy Ghost. 57, a Ghanaian physician, Dr. Konadi Aholo described the AIDS epidemic in the following way. The African does not speak of Africa as if it was a little country somewhere in Timbuktu. Africa is a massive continent with 600 million people and 2,300 tribes distributed in 53 different, sometimes very different, countries. For example, the difference between Ghana and next-door Ivory Coast vis-a-vis the sex trade is the difference between Ghana's ex-colonial master Britain and Côte d'Ivoire's France. Scientific and media descriptions of Africa's AIDS elephant, with its 53 body parts, have sometimes been like those of the proverbial blind men surveying the elephant. Most researchers concentrate on the tusk and, not surprisingly, come out with the AIDS problem in Africa is very sharp and pointed. The whole continent is like that. Even when experts from Nigeria, the large body part of the elephant, confirm with seropositivity studies that there is not yet an AIDS problem in their country, they are shouted down with underreporting. Underreporting. The whole beast has a sharp profile. To these safari experts, Tanzania and Sierra Leone, Uganda and Gabon, Zaire and Ghana, Rwanda and Gambia, are all the same. 58. Dr. Konadi Aholo toured all the AIDS-affected African countries, except Zaire, where he was refused entry, although U.S. government-sponsored AIDS researchers appear to have no such difficulties, and reported his findings in the British Medical Journal and The Lancet. In February and March of this year 1987 I made a 6-week tour of 26 cities and towns in 16 sub-Saharan countries, including those most afflicted by AIDS, did ward rounds with doctors and nurses, met ministers of health, directors of medical services, and research workers, native and expatriate. If one judges the extent of AIDS in Africa on an arbitrary scale from grade I, not much of a problem, to grade V, a catastrophe, in my assessment AIDS is a problem, grade 2, in only 5, possibly 6, since I was unable to obtain a visa for Zaire, of the countries where AIDS has occurred. In no country is the AIDS problem consistently grade 3, a great problem, or ever grade 4, an extremely great problem, and in none can it be called a catastrophe, grade V. In Kenya, for instance, contrary to widespread reports, I would rate AIDS in 1987 as grade 1. Before the days of AIDS in Ghana there was a death a day, on my ward alone of 34 beds. They died from one or another of the following: cerebrovascular accident from malignant hypertension, hepatoma, ruptured amoebic abscess, hematmesis, chronic renal failure, sickle cell crisis, septicemia, perforated typhoid gut, hepatic coma, hemoptysis from tuberculosis, brain tumor, Hodgkin's disease. Today, because of AIDS, it seems that Africans are not allowed to die from these conditions any longer. If tens of thousands are dying from AIDS, and Africans do not cremate their dead, where are the graves? Why do the world's media appear to have conspired with some scientists to become so gratuitously extravagant with the untruth? That was the question uppermost in the minds of intelligent Africans and Europeans I met on my tour. 59. Dr. Konadi Aholo was particularly critical of Western researchers who, with no experience of tropical medicine, used seroepidemiology as a substitute for, rather than an adjunct to clinical epidemiology, and described the difficulties faced by doctors working in Africa who sought funding from external research agencies to increase their clinical epidemiological research base. 60. Although African governments have repeatedly been accused of underreporting and the number of AIDS cases notified to the World Health Organization from African countries have never reached the expectations of the Western AIDS establishment, 53, it is important to appreciate how even these relatively modest figures are derived. In the West, AIDS is diagnosed and hence reported when a patient develops an opportunistic infection or AIDS dementia, 61. The diagnosis is confirmed with at least 2 and often more different types of tests, e.g. ELISA, Western blot, radioimmunoprecipitation assay. Thus the great majority of patients with symptoms and signs of HIV infection, i.e. those with persistent generalized lymphadenopathy or AIDS-related complex, now called symptomatic HIV infection, do not reach the official statistics until they develop opportunistic infections or dementia. There is a degree of underreporting, up to 20% in the United States, but virtually no overreporting, 62, because of the expense of laboratory tests for HIV infection and opportunistic diseases. Physicians and health workers in most African states have been encouraged to use the WHO clinical criteria for AIDS, confirmed with ELISA when available, 63. The WHO clinical criteria do not distinguish AIDS and symptomatic HIV infection, and in Africa both are therefore reported as AIDS cases. 64. Nor do they differentiate AIDS from other clinically similar wasting diseases, and a number of studies have shown that between 26 and 50% of patients who fulfill clinical criteria are seronegative for HIV infection, 65, 66, 67. Diagnostic pitfalls include infections, particularly tuberculosis, parasitic infestations, lymphomas, and occult carcinomas, and endocrine disorders such as diabetes mellitus, thyrotoxicosis, and Addison's disease, 64, 68. Confirmatory testing with ELISA, if available, also presents difficulties, given the high rate of false positivity with this test. In this context, it is curious to note that the proportion of African AIDS patients who have died is much lower than that in the West, where it is consistently 50 to 60%, 64, 65. It is most unlikely that Africans with AIDS live longer than their Western counterparts, and far more probable that reported African cases include patients at an early stage of the disease and patients with clinically similar but less deadly diseases. If the criteria used to diagnose AIDS in Africa were used in the West, the number of Western AIDS cases would increase manifold, and therefore comparisons between the incidence of AIDS in Africa and the West are meaningless. Such difficulties are usually dismissed on the assumption of enormous underreporting of AIDS in Africa, but if this were so, what happens to these patients? Do they die, or do they somehow fade away unmourned, unburied and unrecorded? In Africa as in the West, AIDS is predominantly afflicting the young, sexually active section of the population, and a change in the pattern of disease and death in this group would be reflected in official statistics even if not reported as due to AIDS. This has been demonstrated in Britain, where there has been an increase in the death rate amongst young men, and up to 500 may have died of AIDS in the last year without being reported as such. Yet Western researchers seem incapable of believing that African countries gather such statistical information, although it is often readily available in the libraries of their own institutions. When comparing the incidence of AIDS in different countries, it is also important to consider the rate of progression from HIV infection to full-blown AIDS. It is probable this will be more rapid in countries with a high rate of infectious and parasitic disease, and consequently the proportion of AIDS patients to the number with HIV infection will be higher. Even if African states were using the same criteria to diagnose AIDS as in the West, assumptions about the prevalence of HIV infection based on Western experience would be misleading. Even if one chooses to ignore the information provided by various African ministries of health, some assessment of the scale of the African epidemic can be made by studying expatriate Africans. Many Africans in Europe and America are temporary residents, or travel home frequently, and AIDS in this group should mirror the epidemic in their countries of origin. Whilst there was much excitement about the incidence of AIDS in expatriate Africans in Europe in the early 1980s, 69, 71, the number of Africans diagnosed in Europe actually declined between 1984 and 1986, 72, perhaps because reliable tests for AIDS became available, and only in 1987 showed a modest increase. Africans with AIDS in Europe are no longer reported separately by the WHO, 73, perhaps because they have ceased to be a significant proportion of the total European cases. Although there was much talk of the risks of transmission of HIV-2 by West Africans in Britain, more than 6,500 patients with West African connections were tested and all were found negative for this virus, 74,75. It is curious that expatriate Africans in the United States have never featured in discussions about the supposed African origin of AIDS, nor have they been reported as suffering from AIDS in any number. Sound scientific methodology surely dictates that evidence contrary to a proposed hypothesis should be sought as vigorously as evidence for the hypothesis. In the case of AIDS from Africa, contrary evidence has not been sought at all, but this singular deficiency in effort is then presented as a lack of result. If scientists did wish to explore the possibility that HIV was introduced to Africa from the United States and Europe, we would mention two possible areas for research. The first is the export of infected American blood products. Discussion in the scientific literature about Africa and transmission of HIV by blood products inevitably concentrated on the possible importation of infected plasma to America from Africa, an unsubstantiated hypothesis that died quickly, 76, or the spread of HIV in Africa by local blood transfusions, 77. We could find only one reference to the export of infected American blood to third world countries, in a WHO working paper where it was said that contaminated plasma pools may have been sold at discount prices in developing countries since they could not check the products. 78. Western countries outside the USA are for the most part self-sufficient in whole blood and plasma, and the only significant group infected from America were hemophiliacs who were given imported American clotting factors. Poor countries often cannot afford a blood transfusion service, and wealthy patients with blood loss may be transfused with imported blood whilst the poor at best receive an immediate transfusion from a relative or friend. If imported whole blood was responsible for introducing AIDS into Africa, this would be consistent with the initial appearance of AIDS in the urban-based elite in countries like Zaire, which are particularly dependent on America. It would also account for the development of AIDS in expatriate Europeans, such as the French woman who developed AIDS after a blood transfusion in the Cameroons, as it is unlikely that she was transfused with locally obtained blood. 79. A second, and we suspect far more important route by which AIDS may have been introduced into Africa is sex tourism. AIDS researchers, who seem unable to contemplate that white men can infect African women, have presented AIDS in Africa as a disease transmitted by promiscuous men, and to racist minds all Africans are promiscuous, to prostitutes who then infect foreign clients. 80. Prostitution in African countries tends to occur at two levels, with younger and prettier women seeking valuable foreign exchange who work in the large hotels and night spots which attract foreign tourists and wealthy Africans, and with older and less attractive women whose clientele is predominantly poor and local. If African realities agreed with the researchers' suppositions, older African women and their local clientele would be bearing the brunt of the epidemic, but to the contrary, it is the young women frequenting the tourist centers and foreign military and naval establishments who are developing AIDS and are transmitting it to their African sexual partners, husbands, boyfriends, and wealthy African clients. 58,81. Conclusion. When discussing the issue of the origin of AIDS, we are frequently asked by well-meaning people, does it really matter where AIDS came from? Shouldn't we forget about the origin and concentrate on dealing with the epidemic? Certainly we agree that every effort should be made to contain the epidemic, in Africa as elsewhere in the world, but AIDS researchers have opened a Pandora's box of racism and prejudice that cannot be closed by simply dropping the subject of the origin. Incorrect assumptions about the source and nature of the African AIDS epidemic will also inevitably lead to inappropriate programs for containment and control. Africans have complained that scarce resources from the WHO have been diverted from programs to control major epidemic diseases that are killing many more people than AIDS, and insufficient emphasis has been placed on the risks of sex for money, whilst the dangers of low levels of promiscuity have been exaggerated to such an extent that people have even committed suicide because they feared they had AIDS. Although racism in its various manifestations has come under increasing challenge in recent years, it remains a potent influence, and it is naive to believe that medical science is immune to this particular poison. With the emergence of a new and deadly sexually transmitted disease, it was perhaps almost inevitable that black people would be attributed with its origin and transmission, whatever the evidence. Racism is an irrational system of beliefs without scientific foundation, and much of the confused, contradictory and simply nonsensical conclusions reached by the scientists about AIDS and Africa can be attributed to their attempts to square their research findings with their racist preconceptions, rather than objective scientific reality. The determined pursuit of the African origin has been of little scientific or practical merit, but instead has escalated racism, created conflict between African and Western countries, diverted resources away from areas where they are much needed, and has wasted time. Let us hope we can learn from our mistakes, otherwise we will be doomed to repeat them. Asterisk. End of article. Remarks on methods for retroviral isolation. By Etienne de Harven. Continuum Spring 1998. Dr. Etienne de Harven is Emeritus Professor of Pathology, University of Toronto. He worked in electron microscopy, M, primarily on the ultrastructure of retroviruses throughout his professional career of 25 years at the Sloan Kettering Institute in New York and 13 years at the University of Toronto. In 1956 he was the first to report on the M of the friend virus in murine, mouse, leukemia, and in 1960, to coin the word budding to describe steps of virus assembly on cell surfaces. He will deliver a speech at the 12th World AIDS Conference in Geneva, June 28 to July 3, at the session HIV Testing, Open Questions about Specificity. The most impressive developments of molecular genetics over the past 20 years do not make Robert Koch's postulates obsolete. The first of these postulates indicates that to be considered as pathogenic, a microorganism should be isolated in every single case of the disease. Still, according to E. Papadopoulos et al. And S. Lanka, 2, isolation of HIV from fresh plasma of AIDS patients has never been achieved under any circumstances. Moreover, and most surprisingly, the efficiency of current antiviral therapeutic protocols, AZT tritherapy, is being measured by determining viral load in the plasma of treated patients. Viral load implies viremia, i.e. the presence of circulating viral particles in the peripheral blood. The virus incriminated being allegedly a retrovirus, this would have been the time to remember that the morphology of such viruses in several animal experimental tumors and leukemias had been extensively characterized by transmission electron microscopy, M, over the past 40 years, the viral particles having a characteristic ultrastructure and a diameter ranging between 100 and 120 Some of them had been studied by methods of high-resolution transmission electron microscopy. 3. In the 1960s, transmission electron microscopy was by far the best available method to identify viruses within or around diseased cells. Consequently, many cancer research centers all around the world started to compete for the best equipment and training in M, aiming at the demonstration in human malignancies of viruses similar to those which had just been recognized as significantly associated with tumors and leukemias of several laboratory animals. This approach to cancer research appeared highly justified when Lewoffe, Horn, and Tournier proposed to classify all viruses primarily on the basis of their morphological features demonstrated by electron microscopy. 4. Identification of viruses by M in leukemic animal tissues became unambiguous when steps in virus assembly, i.e. the budding of complete virions from the surface of the infected cells, were described. 5. In spite of considerable efforts, the search for similar, typical viruses in human malignancies remained entirely negative. Pleomorphic membranous microvesicles, approaching viral size, and frequently described in the literature on human malignancies as virus-like particles, were without any pathogenic significance. As stated in 1965, typical RNA tumor viruses have never been observed in association with human neoplasia. 6. Concentrations of retroviruses from murine and avian leukemic tissue homogenates were reproducibly achieved, permitting titration of infectivity into receptive laboratory animals. This was not, however, an easy approach to the problem of virus purification, large amounts of microvesicles and cell debris being usually present. As far as virus purification was concerned, it soon became evident that when viremia is present, blood plasma was far better than tissue homogenates for efficient virus isolation and purification. In the case of RNA tumor viruses, now called retroviruses, the demonstration of viremia in the blood plasma of experimental leukemic animals, chickens and mice, was published more than 35 years ago. A most efficient purification method including ultrafiltration and ultracentrifugation of a 1/1 dilution of plasma in heparinized Ringer's solution, allowed me to demonstrate packed retroviruses by transmission electron microscopy. 7. In thin sections of pellets obtained by high-speed centrifugation of the purified virus, quite clearly establishing that the amount of contaminating cell debris was remarkably small, a conclusion which could never have been reached by using the negative staining method. Using this simple ultrafiltration procedure, virions were never exposed to hypertonic shock. However, sedimentation in sucrose density gradients, at the density of 1.16 gm/ml, soon became the most popular method for retrovirus purification. 8. Interestingly, it was very well known by electron microscopists in the 1960s, that sharp bands sedimenting at the density of 1.16 frequently contained large amounts of microvesicles and cell debris of non-viral nature. These debris just happened to sediment in sucrose gradients at a density very similar to that of retroviruses, clearly indicating that finding a sharp band at the density of 1.16 gm/ml was of little significance and was certainly far from any demonstration of retroviruses isolation. But this conclusion was based on M findings, and around 1970 the faith in retroviral oncology was assuming quasi-religious proportions. If M cannot demonstrate viruses in the 1.16 bands, let us forget about M and rely on other markers. When around 1980, R. Gallo and his followers attempted to demonstrate that certain retroviruses can be suspected of representing human pathogens, to the best of my bibliographical recollection, electron microscopy was never used to demonstrate directly viremia in the studied patients. Why? Most probably, M results were negative and swiftly ignored. But overenthusiastic retrovirologists continued to rely on the identification of so-called viral markers, attempting to salvage their hypothesis. When retrovirus particles are legion, the study of molecular markers can be useful, and provide an approach to quantification probably better than direct particle counting under the M, which I always found very difficult. But when, using M, retrovirus particles are absent, relying exclusively on markers is a methodological nonsense. Markers of what? Nevertheless, for the past 10 years, HIV research and clinical therapeutic trials have been primarily based on the study of several HIV markers. First, the antibody ELISA, then Western blot tests were hastily developed, at sizable financial profit eagerly split between the Pasteur Institute and the U.S. Seropositivity became synonymous with the disease itself, plunging an entire generation into behavioral panic, and exposing hundreds of thousands of people to preventive antiviral AZT therapy, which actually hastened the appearance of severe or lethal immunodeficiency syndrome. Appropriate controls were apparently never carried out or were never published. Still, back in 1993 it became clear that the so-called HIV antibody tests badly lacked specificity. 9. Cross-reactivity being observed with patients suffering from a long list of pathological conditions including malaria, leprosy, autoimmune diseases, and many more. Secondly, viral proteins. Several proteins have been identified as HIV markers, most frequently because they were identified in a variety of 1.16 bands. The case of the P24 viral antigen is a significant example, and its lack of viral specificity has been well documented. 10. Third, reverse transcription. If reverse transcriptase activity were a unique feature of retroviruses, it could have been an interesting molecular marker. Unfortunately, it has been shown that reverse transcriptase is found in the uninfected cells of yeasts, insects, and mammals. 11. And has nothing to do with retroviruses as such, as well referenced in a recent report from S. Lanka. Moreover, Camillus himself does not support the use, to amplify and quantify the HIV genome, which is being made of the PCR methodology he developed, which is the current method of measuring the viral load in AIDS patients. More disturbing is the fact that some markers are searched for in the 1.16 gradient sedimenting material, which is the density where intact virions are expected to be found, but not their molecular fragments. If lysed retrovirus particles released molecular markers, the 1.16 samples should at least initially allow investigators to demonstrate virus particles by M. They don't. However, after 15 years of most intensive HIV research, 2 independent groups finally decided to explore by electron microscopy the ultrastructural features of the material sedimenting at the 1.16 density. Working on HIV-1 infected T-cell cultures supernatants, both groups found that it contains primarily cellular debris and cell membrane vesicles which could definitely not be identified with HIV particles and rare virus-like particles. 12.13. Still, this is the type of sample in which viral markers are currently identified and used to measure the effects of antiviral drugs in current clinical trials. In conclusion, and after extensive reviewing of the current AIDS research literature, the following statement appears inescapable: neither electron microscopy nor molecular markers have so far permitted a scientifically sound demonstration of retrovirus isolation directly from AIDS patients. This conclusion fully confirms the recent reports published in Continuum by E. Papadopoulos and by S. Lanka. Harvey Bialy, editor of the journal Bio/Technology, has stated that, 14, a powerful hypothesis has to explain and predict. What kind of scientist continues to support a hypothesis that fails to explain and fails to predict? The HIV/AIDS hypothesis fails to explain the considerable drop of T4 circulating lymphocytes in AIDS patients. It predicted a dramatic AIDS epidemic which was never observed, unless we accent the CDC's most surprising redefinition of AIDS as including some 31 AIDS-defining illnesses. Obviously, the HIV/AIDS hypothesis has to be scientifically reappraised. 15. And, most urgently, the funding for AIDS research should no longer be restricted to laboratories working on an hypothesis which has never been proven. Asterisk. End of article. Retroviruses. The Recollections of an Electron Microscopist. By Etienne de Harven. Reappraising AIDS November/December 1998. For an electron microscopist who spent most of his research career studying retroviruses associated with murine leukemias and who followed closely their hypothetical impact on the field of human oncology, it was predictable that contemporary AIDS research was completely on the wrong track. The following recollections are presented to explain why. The importance of electron microscopy in the emergence of modern cell biology, between 1945 and 1965, is unanimously recognized. Unquestionably, the relationships between cell structures and cell functions would never have been elucidated without the high resolving power of the electron microscope, M. What is less generally recognized, however, is the role virus research had in the study of cell ultrastructure. Historically, when Rudenberg filed the patent for the electron microscope in 1931, 1. His motivation stemmed from the hope of visualizing the polio virus. And during the WW2 years, when electron microscopes started to be accessible to biologists, attempts to visualize virus particles associated with cancer cells of laboratory animals received first priority. Albert Claude, working at the Rockefeller Institute, succeeded in demonstrating the Rous sarcoma virus in chicken fibroblasts, 2. And a few years later, Keith Porter et al. Had a similar success in imaging the milk factor in murine mammary adenocarcinomas cells, 3. The viral etiology of Rous sarcoma in chickens and of mammary tumors in mice were well established by microbiological ultrafiltration experiments, years before the M pictures were published. Still, the direct observation of virus particles in these experimental tumors gave an enormous, today, we would perhaps say excessive, impetus to virus research in oncology. Viral etiology of several murine and avian malignancies had been clearly demonstrated by ultrafiltration experiments which permitted the calculation of the approximate diameter of the virus particles. Electron microscopists knew, therefore, what size particle to look for, frequently around 100 nm. This facilitated the initial identification of oncogenic viruses by M, although it was later observed that many microvesicles or particulate components of normal cells fall into a similar size range. The discovery by Charlotte Friend, at the Sloan Kettering Institute in New York, of a murine erythroleukemia transmissible by cell-free filtrates illustrates well the research approaches around 1955. Moreover, since I started working in Dr. Friend's laboratory at that time, the rationale of our research program is clearly in my mind. For M, we gave priority to two types of sample: 1, a variety of tissues from leukemic Swiss mice, spleen, lymph nodes, thymus, and bone marrow, and 2, ultracentrifugation pellets obtained from leukemic tissue cell-free filtrates which readily transmitted the disease to adult Swiss N/or DBA/2 mice. We knew from ultrafiltration experiments that the activity was absent when using filters with an average pore size diameter smaller than 200 nm. Classic theories on ultrafiltration indicated that the infective particles were therefore probably in the 100 nm range. The study of thin sections from plastic embedded leukemic tissue frequently revealed the presence of particles of approximately that diameter, closely associated with a variety of cells. The particles were bound by a single membrane and had a centrally located electron-dense core or nucleoid. They had a characteristic structure and strikingly constant diameters. To our knowledge, such particles did not resemble any known components of normal cells. However, they did resemble particles previously identified by others in several filterable experimental tumors and classified by W. Bernhardt as type C particles. 4. More importantly, we observed identical particles in ultracentrifugation pellets prepared from cell-free filtrates capable of transmitting the disease to susceptible mice. On the basis of these data, we hypothesized that these particles indeed represented the oncogenic virus etiologically related to the friend erythroleukemia. 5. Somewhat surprisingly, the virus was also found in close association with cells not apparently related to the leukemic process, such as bone marrow megakaryocytes, for example. These early M studies also indicated that all electron-dense particles averaging 100 nm in diameter are not viruses and that a rigorous ultrastructural characterization was essential for adequate differentiation between viruses and virus-like particles. Fortunately, our M studies of friend leukemia soon added an important feature to the structural characterization of oncogenic RNA viruses. It appeared that virus assembly is a cell surface phenomenon, the surface membrane of the infected cell directly contributing the future viral envelope by a multiple-step mechanism for which we coined the word budding. 6. Viruses are released into intercellular spaces by such a budding process. M identification of viruses of that group in other experimental malignancies became therefore more rigorous, observation of budding particles being required. This probably helped in eliminating thousands of virus-like particles observed in human malignancies and with which overenthusiastic electron microscopists attempted to contaminate the literature. In addition, recognition of budding particles on cell surfaces clearly allowed us to identify infected single cells, and to observe that such cells are perfectly viable, with absolutely no ultrastructural evidence of any cytolytic effect of viral infection. Moreover, typical viruses were frequently observed in cells undergoing mitotic division. 7. Since obviously human experimentation was out of the question, the eventual observation in human cancer cells of particles resembling those well characterized in experimental tumors would have been of great interest, although by itself not conclusive. Around 1960, many laboratories around the world, using ever-improving M methodologies, were focusing on that target. At that time, well before the emergence of molecular biology, M was the best we had to identify viruses in cell samples. The crucial role of M was well recognized at the Cold Spring Harbor Conference of 1962, when Lwoff, Horn and Tournier proposed classifying all viruses primarily on the basis of their morphological characteristics demonstrated by electron microscopy. 8. Continuing our studies on the friend leukemia virus, FLV, and encouraged by Dr. J. Beard from Duke University who had considerable experience with avian leukosis, we oriented our effort toward the demonstration, by M, of viremia in leukemic mice. The most efficient initial step in purifying avian leukosis viruses was to start not from leukemic tissues but from the blood plasma of leukemic chickens. We wondered if a similar situation could also prevail in leukemic mice. This was important for us because the efficiency of our early virus purification method from homogenates of leukemic tissues such as spleen or lymph nodes was poor. We developed, therefore, a very simple purification procedure based on 2 steps of millipore filtration. A diluted plasma sample, around 10 ml, i.e. bleeding about 25 leukemic mice, was first clarified by aspiration filtration through 0.65 filter. Filtrate number 1 was then further filtered through a 0.22 membrane. Filtrate number 2 was then centrifuged at 30,000 grams for 120 minutes. The resulting pellet was extremely small, almost invisible, still worth embedding for M. Thin sections of such pellets revealed the presence of a most impressive population of typical, well-preserved virus particles, packed together, and with very little contaminating debris. 9. This was our approach to the demonstration of viremia in 1965. Meanwhile, many M cancer research centers, those led by Drive W. Bernhardt, at Villejuif, France, Dr. A.J. Dalton, at NCI, Bethesda MD, Dr. Eld Mohovsky, at MD Anderson, Houston, TX, and us at Sloan Kettering, New York, were spending a considerable amount of time in attempts to demonstrate virus particles associated with human cancer cells. Virus-like particles were occasionally reported but convinced nobody. Typical viruses were never conclusively demonstrated. This was in sharp contrast with the highly reproducible demonstration, by M, of viruses in a variety of murine and avian leukemias and tumors. Very few papers were published to report on these negative findings in human cancers and leukemias. However, Hogna, in 1959, 10, reported on the difficulty of identifying any typical virus particles in a large series of human mammary carcinomas. Bernhardt and Laplace, in 1964, 11, in an M survey of cases of Hodgkin's disease, lymphosarcomes, lymphoid leukemias, and metastatic diseases failed to recognize virus particles associated with these malignant conditions. At Sloan Kettering in New York, I decided, in 1965, to stop surveying cases of leukemias and lymphomas by M for the presence of viruses in view of our entirely negative results. This was reported at a conference on methodological approaches to the study of leukemias held at the Wista Institute, in 1965, 12. Of mice and men. Publication of these negative findings failed to discourage fanatical virus hunters. An explanation for these negative results had to be found somewhere. Perhaps the technique of M by the thin section method was not the best approach? Although it worked perfectly for mice, preparing thin sections was time-consuming and skill-demanding. Who had time for that, when research funding was getting difficult, and when major pharmaceutical corporations were starting to finance crash programs for speedy answers? Why don't we try the negative staining method? It is very easy and very fast. And, after all, it gave beautiful results with unenveloped viruses like adenovirus and polyoma. The results were absolute disaster because fragile RNA tumor viruses, not yet called retroviruses, are badly distorted by air drying during the negative staining procedures. They appear as particles with a long tail. Unfortunately, many cell debris and vesicular fragments, when air dried for negative staining, form similar tailed structures. Interpreting tailed particles as RNA tumor viruses was therefore a bonanza for virus hunters. Still, we had demonstrated that tailed virions were preparation artifacts which can be avoided by proper control of the osmolality and by osmium fixation prior to negative staining, 13, or by critical point drying, 14. The chaos created by reports on tailed particles damaged the credibility of M in the search for cancer-related viruses. Cow's milk and human milk were being screened for tailed particles, and Sol Spiegelman was eloquent on the possible risks of breastfeeding. A major discovery, which had nothing to do with M, completely reoriented ideas on how RNA tumor viruses might work, the discovery by Temin and Baltimore, in 1970, of reverse transcriptase, RT. How RNA tumor viruses could induce genomic alterations of the cells they infect suddenly became comprehensible. Moreover, these viruses remained candidates as possible oncogens because they were known not to be cytolytic. RNA tumor viruses were given a new name, retroviruses, and the study of their eventual role in causing human cancer soon received levels of federal funding, after Nixon's War Against Cancer Act, which were frighteningly in excess of what could have been expected from a frequently recurring, perhaps interesting but so far totally unproven hypothesis. The profile of the research effort changed very drastically after the discovery of RT in 1970. Somehow, most of the research methods which had dominated the field of viral oncology from 1950 until 1970 were suddenly substituted by the most exclusive fashion of molecular biology. I observed this evolution almost as an outsider, since, in my view, electron microscopy was no longer a primary contributing method to tackle any further the hypothetical relationships between retroviruses and human cancer. The 1970 to 1980 years were dominated by a series of ideas which would never have withstood scientific scrutiny 10 or 20 years before. For example, 1. It became acceptable to postulate that when viruses cannot be seen by in cancer cells, biochemical or immunological methods supposedly identifying viral markers were enough to demonstrate viral infection of the cells under scrutiny. Such markers can be an enzyme, RT, an antigen, various proteins, or some RNA sequences. Never seeing the viral particles was conveniently explained by the integration of the viral genome into the chromosomes of the alleged infected cells. To accommodate such interpretations implied complete oblivion of all we knew from previous research on cancer of experimental animals. Admittedly, in these models M was only showing terminal steps of viral reduplication, initial phases being a series of molecular events which escape ultrastructural recognition. Still, in all the classical models such as murine and avian leucoses, visible terminal steps of viral replication, i.e. budding, were always observed and regarded as essential for the spreading of infection from cell to cell. Another shortcut with disastrous consequences has been the naive notion that any material banding at 1.16 gm/ml represented retroviruses. Sure enough, true retroviruses band around that density. But this does not mean that whatever material bands at 1.16 gm/ml is retroviral in nature. In the 1960s I was frequently asked to look at such 1.16 gm/ml bands by biochemists. Look at this, it forms a sharp band, it should be pure viruses. Ultracentrifugation pellets obtained from such sharp bands showed, in thin sections for M, an extreme variety of microvesicles and proteinaceous debris, but no retroviruses. Still, this approach has been, and still is, used to identify virus markers. How sad it is to think that a simple M control of such bands, which takes about 2 days, and costs a few $100s, but has never been done before 1997, could have prevented these highly misleading interpretations of markers on which large budgets have been simply wasted. 3. Collecting viruses from the supernatant of cultures of virus-infected cells raises other questions. We all remember the discovery, by Epstein, 15, in 1964 of the EB virus in cultures derived from African Burkitt's lymphomas. This was an M finding and the virus was immediately and properly classified as a member of the herpes group. To identify this DNA virus in cultured cells it was necessary to look into partially degenerating cells because, most obviously, the virus had a marked cytolytic effect. By complete contrast, retrovirus-carrying cells maintain excellent viability and released viruses can be easily recovered in the culture supernatant without the need to apply any lymphokine or growth factors stimulation to the cultures. For as far as scientific policy is concerned, research on potentially oncogenic viruses was dominated by the retrovirus hypothesis. Federal funding took the same direction, amplified by the incredibly naive idea that success was primarily a matter of money. Unusually large levels of federal support resulted in the creation of a retrovirus research establishment. Large numbers of research jobs were created in this venture. The intellectual freedom to think along other avenues of cancer research was rapidly dwindling, especially when major pharmaceutical companies started to offer tantalizing contracts to support polarized retrovirus research. The top priority was to demonstrate, at any cost, that retroviruses had something to do with human cancer, an hypothesis, however, which didn't receive the slightest support throughout the 1970s. Such a misdirected research effort would have been relatively inconsequential as long as public health was not involved. Unfortunately, the emergence of acquired immunodeficiency syndromes, AIDS, in 1981 gave the retrovirus establishment an opportunity to transform what could have been only an academic flop into a public health tragedy. What happened after 1981 is so well known to all the readers of RA that I hesitate to elaborate any further. The events which have led to today's crisis have been reviewed and analyzed most convincingly by Peter Duesberg, 16. I must say that I read Duesberg's book with great attention but basically with no surprise, because the way research had been conducted on retroviruses in the 1970s had so dangerously set the stage for impure science. 17. Soon after the first cases of gay-related immune deficiency were described by Michael Gottlieb, it was obvious for all observers that Gallo and his associates were going to jump on the new syndrome as a God-sent opportunity to attempt to justify the lavish federal budgets they had consumed on retroviruses over the past 10 years. In 1980, the scientific community was getting more and more concerned about the absence of results in the war against cancer based on retrovirus hunting. The minor episode of HTLV1 was not enough, by far, to calm the fears of grossly misdirected federal research funds. The fact that the syndrome, soon tactically renamed AIDS, had nothing to do with cancer was apparently of little embarrassment for Gallo. Frequent association with Kaposi sarcoma helped to blur the difference in the eyes of the public. Dominated by the media, by special pressure groups and by the interests of several pharmaceutical companies, the AIDS establishment efforts to control the disease lost contact with open-minded, peer-reviewed medical science since the unproven HIV/AIDS hypothesis received 100% of the research funds while all other hypotheses were ignored. The general public and the medical community were made to believe that the presence of circulating antibodies is diagnostic of this disease, that Koch's postulates were outdated, that 90% of all cases of an infectious disease can be observed in males, that viremia can be measured by PCR enhancement of RNA fragments even when viral particles are not demonstrable, etc., etc. Most conveniently, it was totally forgotten that heroin addicts were known for many decades to expose themselves to immunodeficiencies, that nitrite inhalants have multiple toxic effects, that the extreme toxicity of AZT was known for over 20 years, that known retroviruses never have any cytolytic effects, etc., etc. And to ensure that the AIDS establishment could profitably continue to flourish, research on any dissenting, i.e. non-HIV, hypothesis was carefully prevented by tight control of research funding and by the extreme difficulty of publishing anywhere any dissenting views. In the late 1980s, I was considering adding to my research program in Toronto more observations on samples from AIDS patients. Unfortunately, by that time the media and the CDC had so perfectly orchestrated the panic of a plague-like epidemic that I was quickly made to understand that my assistants would all transfer out of the lab if I had insisted to activate such a program. The HIV seropositivity test was still at that time regarded as providing reliable diagnostic data. Since then, Papadopoulos and the Australian team have demonstrated that this is very far from the truth. 18. Since my retirement in France, I take any opportunity to speak out, as openly as I can, along the lines of this little note. I am proud to be part of the group for the reappraising of the HIV/AIDS hypothesis, and I sincerely hope that the combined activities of the group will soon contribute to a complete reappraisal of the etiology of AIDS for the best interest of patients' care, and for a revival of scientific integrity in medical science. Copyright 1998 by Professor Etienne de Harven, M.D., L.E. Mas Patou, 2879 Route de Grasse, 06530 Saint-Cyger-sur-Siagny, France. Patou.de Harven at wanadoo.fr. End of article.